Yes, thank you. Thank you very much for your kind introduction, and I will happily address this exciting conference. A very good morning to you all, speakers and delegates and student participants. And I would like to express my welcome to the global webinar on material science and engineering. Today with the subtitle Synergic Growth and Advancements in Material Science and Engineering. My name, as was just stated, is Dr. Per Arvid Luffman. I'm from Biotech company in Hamburg, Germany, as well as the Kaiserslautern University of Applied Sciences and the Hamburg University. I am the chairperson of this conference, and I am happy that we can all meet in this webinar internet platform setting again this year. Yes, even though the worldwide COVID-19 pandemic is still far from over yet, I think we all try to as good as we can to continue our international and global scientific exchange. And this webinar is a very good example of this continuation within the pandemic, which I think is a very an extraordinary good thing to do. Me as a multidisciplinary material scientist, as a multidisciplinary researcher, I am very happy to open this webinar, which this year concentrates on synergic growth and advancements in material science and engineering, as I was just pointing out. This time we are fortunate enough to take part of exciting multidisciplinary presentations covering very diverse fields such as nuclear waste, bioactive films, cement industry, stem cells, atomic scale, fracture analysis, hypothetics, aluminium alloys, and even materials stemming from the Biosphere, if I humbly may mention my own tiny little contribution to this webinar. As we can see by this, all the different kinds of fields that I was just listed, that I just mentioned, material science remains an exciting field for the future. And this webinar clearly reflects its subtitle, Synergic Growth and Advancement in Material Science, and that we can also expect this as an outcome of this webinar. So please do not hesitate to address your own thoughts and ideas throughout the conference, and let us together make it a memorable event that we would like to come back to again and again and again in the future. On behalf of the organizing committee, it gives me great pleasure to welcome you to our meeting organized by the Global Scientific Guild. Thank you all for attending the conference today and tomorrow. It marks an important event initiated by the Global Scientific Guild. This will provide the opportunity to share research findings and encourage new collaborations, and to interact in various areas of material science and engineering. The global webinar on material science and engineering is a scientific congregation that brings together researchers, scientists, key decision makers, industry professionals in the same physical, or this time I would rather say virtual space, for a brief yet intense period of discussion, collaboration, and generation of innovative solutions to address overarching challenges related to the theme of today, synergic growth and advancements in material science and engineering. We are very pleased to welcome you to both share and witness advances in this field. The conference has attracted speakers and delegates from all over the world. I would like to thank all speakers and event partners for their continuous support and patronage. The Global Scientific Guild also wishes to thank all the organizing committee members who have put in long hours and generated many creative ideas to bring this meeting together. Finally, we thank each and every one of you, the participants at this conference. Again, a warm welcome to all webinar participants, and please enjoy the global webinar on material science and engineering, synergetic growth and advancement in material science and engineering. Thank you very much.
Thank you, Dr. Luthman, for the introduction speech. And
usually, if we have a conference face to face, we do something like this, so we can do it.
thank you.
Very good, very good. I support that.
Yes. Usually I think that this part is missing for of all of us.
Oh yes, oh yes. We don't interact, and we don't show. It's harder to show feelings and excitement when you are online. But, it is a good initiative.
Yes, okay.
Very much.
Thank you. Thank you, Dr. Alina.
Yeah.
And moving on, let's begin the talks. Our first speaker for today is the chairman himself, Dr. Per Arvid Luthman from Apatite, Germany, and he's going to give a talk on nanomaterials in the Biosphere. And I request Dr. Luthman to start his presentation.
Yes, thank you very much. I just have to make sure that you can see my presentation also. Let me make sure that, please let me know if you can. Let me see this.
Yes. Yes.
I guess you can see my presentation now.
Yes.
Right?
Yes. Please.
Okay. I'm happy to hear that. So, yeah, thank you very much for organizing this conference. And I'm going to start, obviously, this conference with this somewhat maybe unusual topic, but I hope that you will get a little bit insight into this. What I'm talking about are nanomaterials in the biosphere. So first of all, we have to clarify what is the nanosphere. Excuse me, what is the biosphere? Nanomaterials, I think we all know. What I'm going to talk to a lot about today is basically building blocks coming from the biosphere. And the biosphere is the sphere where viruses live. We cannot say that viruses live because they don't. They are not alive. But I'm going to address a little bit. I might just move forward one step. I will show viral building blocks that can be put together in various ways to build up nanomaterials. And as I said, the building blocks comes from the biosphere. Then the biosphere is a part of the biosphere. I will go into this a little bit more. This is maybe a little bit fundamental talk that I'm going to hold today, but I think it could, provide a little bit insight and also outlook for future novel endeavors. So I will talk about the biosphere. I will also go into the overarching theme in a way, which is the biomolecular architecture. That is to say, to build with biomolecules. Viruses are by definition nothing less than super huge aggregates of biomolecules. I will go definitely, especially into Archaea virus and Bacteriophages. Those are viruses that infect Archaea and bacteria. I will talk about the nanomaterials, and I will also at the end talk about more about the other things that by definitions are not virus, right? But they still belong to the biomolecular architecture scope of building new materials. I'm talking about DNA. I'm now also talking about HAMI and other appendices. Appendices, what is that? That is something that appendices on the cell surface of microorganisms that can be used for various tasks at the nanoscopic level. So let's start and go into the basics. So this is a sphere. It's our Earth. It is a round little sphere, and this sphere belongs to, or to this sphere, we have different other spheres like the atmosphere, cryosphere, hydrosphere, biosphere, and so on. The biosphere is the place where living things are, and hydrosphere is where you can find water. Cryosphere, there we are talking about the North Pole and the South Pole. Anthrosphere, that is of course the place on Earth where we humans live. So it's quite easy. The atmosphere is also, well, basically the gaseous part of the Earth, if you so wish. Air, etc. And before I continue, I might just look into this level of organization a little bit. We are in this, when you were talking about viruses, etc., we are within this part of the organizational level: ecosystem, communities, populations, and organisms. If we look at the organization of the reality, if you so wish, we go from universe, galaxies, solar system, planets, and then we have the ecology, even all the way down to atoms. So we are going to talk about cells down here, and also everything that belongs to this ecosystem. And so the biosphere, that's where we are. It consists of the atmosphere, the ecosphere, hydrosphere, and lithosphere. This is the place where living organisms live on the Earth. This, I am going very much from a very big perspective, but I think could be important to do so, because in this way you can also get a little bit feeling for where can we do material science, where can we find nano materials building blocks. So this is the biosphere, a nice sphere made by the artist Dick Termes. I just feel free to show this to you because I think it's so beautiful. You see the DNA molecule here, you see a cell membrane, an embryo, and so on. So this is Dick Termes' interpretation of the biosphere. Very beautiful. Here is the virusphere. The virusphere is very simple. It is the sphere of the part of the biosphere that contains viruses. And today I'm going to concentrate on two of those cones here, not all the three of them. Bacterial viruses, they infect bacteria. Archaea viruses, they infect archaea. What does it mean? Archaea, I don't think I have to explain this, because we are in a pandemic. We are dealing with infections every day. Everyone knows what a viral infection is, sadly, but I think that's what we actually do. The bacterial viruses, they are called bacteriophages. Archaea viruses are called archaea viruses. It is as simple as that. Archaea, what is that? Maybe most of you don't know what it is. I tell you what it is. It is basically microorganisms that look like bacteria, but by definition, especially genetical, does not belong to the group of bacteria. I think for material scientists, I think we don't have to take it further than that. I will show you more about this in just a second. Now, before continuing, I will also point out one thing which is important, not only in terms of biology, but or microbiology, but also in terms of material science. And let me explain this. What I'm looking at are hyperthermophiles. Those are microorganisms that can live and strive above 80 degrees C. And bacteria, some of them can live at such high temperatures as 95 degrees. Archaea can even live up to 113 degrees C. We're talking about those microorganisms that live in hot wells and things like that. So we are dealing with hyperthermophiles. Why is this this group important or interesting also for material science? It is because if we look at those proteins and lipoproteins and other biomolecules that those entities have, including their viruses, I must say, we are now talking about the hosts, the bacteria and the archaea. They can live and survive at such high temperatures high temperatures in terms of compared to other biological materials, right? Something is stable here. We have all learned in school at some point of time that proteins, etc., they denaturate and they cannot survive at 130 degrees Celsius. Here they can. So there's still a lot to learn about this field, but what we can say so far, taking it from a material standpoint, this is something stable here. We have biomaterials that are stable at higher temperatures, and that is a good reason for looking into this much more in detail. They, so to say, fulfill the prerequisite for providing stable biomaterials. So let's continue. As I said, I will continue. I will also talk about those particular entities as building blocks, and the building blocks in a concept that I would like to call biomolecular architecture. Let me just go shortly into this. I think you know much of this, that what and how. We are talking about, of course, top-down approach and not about the bottom up, excuse me, the other way around, bottom up and not the top down approach. In this case, with biological entities that self-assemble, they also find each other, if I put it, simplify it like that, via molecular recognition, that is to say, antigens, antibodies, receptors, etc., connect to each other and build up materials in such a way. DNA is another example of this, bacteriophages and viruses, as I mentioned, but you can also talk about proteins, lipoproteins, and so on. I will also mention molecular biology and some cloning. You can, by cloning, introduce the proteins that you would like to have at the ends of a bacteriophage, for example. Architecture is basically the concept that I'm looking at when I would like to go into how we can do with this. By controlling the connection between those entities, we can do architecture at the biomolecular level by introducing functional groups at the end of those entities, and it can be any entity. I'm talking about bacteriophages, but also other entities. And we can build up different kinds of structures, and I will show you some example of those later on. It can be, the more we can control the way how those building blocks can bind to each other, the better we can make architecture. And at the end, we might even be able to do something like this, the Eiffel Tower in Paris. So here I mentioned some examples. This is a Acidophilus bottleneck virus. This is an Archaea virus, which looks like a bottle, and we can very well use this as a nano container, filling the inside of this bottle. It really looks like a bottle. So this is a nano container coming from the Archaea sphere. As well as my favorite, this is an Archaea virus called AFB1, Acidophilus fibreus virus 1, and it connects via, here you can see that at the bottom, via a claw mechanism to pili that extends, a cell appendage that extends from the cell. We even have SERV2, which is a rigid Archaea virus, very good for nanomaterials. We have lemon-shaped viruses that actually can, according to pH and temperature, they can extend their, they can change the length here in this direction, right? With only 10 nanometers, I think it is. So here we have a very precise nanomaterial that can fluctuate in very tiny dimensions, 10 plus minus 10 nanomaterials, depending on pH value. That is a very precise nanomaterials reaction. Here is even the, I'm not sure how much I should bore you with biological things here. It's a little bit, this is typical biological, you know, biologists, they like to organize everything. So let me just yeah, how should I put it? Let me just mention this quickly. Here we are talking about some classes of viruses. For example, the ones that I mentioned, they belong to the Crenarchaeota virus family here. The ones that I showed you are Ampullavirida and Lipotrixvirida, as well as Rudevirida. Not so important to remember. It's not going to be a test after this talk, so don't worry about it. So this is the way, this is SIRV2, the rigid one that I mentioned, the AFV1, as well as the bottleneck virus. The AFV1 virus is interesting to me because we have a claw at the end, and with this claw, we can, via molecular recognition, build up entities. It looks like this. It has only like a 20 nanometer opening, which makes it a wonderful nanomaterial, wonderful viral nanomaterial for constructing novel nanomaterials based upon viruses. And that is much about what I'm going to talk about here, or continue to talk about, how we can connect materials, building blocks at the how we can connect viral building blocks at the nanoscopic scale. So it's truly, as my talk is called, nanomaterials in the Biosphere. Here it's the same picture again. You can see that this is, I should also point out, this is the first stage of infection. This virus connects to this pilus, which is connected to the cell surface. So the first stage of connection is molecular recognition by this virus, and then it enters the cell. Now, as a material scientist, we don't think about infections, we think about building. So with this is a very specific connection, and we can build up materials, consisting of the pili as well as the virus in sort of a mesh network. And this is something very interesting because it is so specific. It doesn't matter how much junk you have in the surroundings. This virus will always, always, always connect to this pilus. I will tell you more about this in a second. Yeah, so this is more examples of the same thing. You can also see the big claw here trying to connect to one other virus, which may also be an alternative way of connection. Yeah, those are more pictures of this. Here we can, when I do research in this field, I also try as much as possible to extend this research into connections between the biological and the inorganic world. In this case, I have made a couple of experiments where you coat the nickel nanorods with the specific protein coming from the pilus and make it connect to this claw that you see here at the end, right? And with this, in this very simple method, you can connect inorganic nickel nanorods to biological viruses. And in this way, you can step by step, bottom up, build up nanomaterials from the Biosphere, and we can extend this even more. We can use other viruses to connect with this claw, carbon nanotubes and other nanorods. So this is a field where we can extend our basic connection mechanisms, and how should I put it? We can. This is the start of nanoscopic building up of materials, right? So yeah, I maybe mention very quickly some very experimental parts. Here you can see an ultracentrifugation tube. It's not so important maybe, but I just mentioned this quickly so that you guys get an idea about where do those viruses come from and how do you get them? How can you isolate them? So you can, first of all, what you do is that you ferment them, and then you isolate them and separate the entities with ultracentrifugation. Ultracentrifugation is basically very, very, very high speed centrifugation. We all know centrifugation. And then you take out, oh, here you have a good pictures. We have different kinds of bands, and those bands, this is a tube after ultracentrifugation. With those syringes that you can see here, you can pull out the different kinds of bands, as you see here, and they belong to, as in this particular case, at the bottom here, they're basically just junk and cells and debris. In the middle band, you see a high density of viruses, and in the upper part, you can also see viruses and pili. So just wanted to mention this briefly, not that we have to go through all the different kinds of laboratory procedures, but maybe it is a good idea to take a little quick look into where do we get those little guys from. Claw attachment is something that I'm looking into increasingly. As I said, it can be induced by external changes or molecular recognition. It is, I believe, more in the second part here, the second part of the hypothesis, excuse me. This is still a matter of research. A lot of research has to be put into this in order to make it more refined. What we see also, if we look at this is maybe not a very good picture, but and there are other ones that if I make a sketch of it, you can see something like this. You have a protein. This is, by the way, this is the cloth that I was talking about. You seem to have a region of protein. You can see this clearly in STM pictures because the different kinds of how should I say, the different kinds of well, color or yeah, let's call it that. The different kinds of how brightness corresponds to different proteins. Protein A seems to be at the very end, protein B, protein C, and then we also at the bottom have some kind of rings or globular structure, globular proteins. Now, the real mechanism of why and how this thing is closing, if it is closing itself or if it is simply just the shape stays the same and it simply just connects via molecular recognition. That we don't know yet, but that is a matter of research that I am happy to be part of. So this is the way this claw looks like. And again, this is a way of organizing materials at the nanoscopic level. Bacteria, yeah, so that was basically what I wanted to tell you about the Archaea and Archaea viruses. Would like to continue a little bit. There's a lot more to say about this, right? But I think I would make the whole thing a little bit more complete and hurry up by also addressing bacteriophages. Bacteriophages are, as I told you, they're simply just viruses that infect bacteria, and they can be rod-shaped. This is a rod-shaped, a model of a rod-shaped bacteriophage, which is called M13. It has a lysogenic reproduction cycle. You don't have to know anything about that, but I tell you anyway. It means that the virus does not kill its host cell, quite in contrary to lytic reproduction cycles. Now, we don't have to go into the details here, but I would like to point out that this bacteriophage M13 is a sort of working horse. It can easily be used for molecular cloning, and that means that you can express, by the way, I should point this out, P8, P7, P9, and P6, and so on, are simply just different kinds of proteins that you can see in the structure. So, you can express whatever kind of protein you want, almost, for example, at the end. So you can induce a much better binding between the ends, and with this, you can do biomolecular architecture. Let me show you a little bit of this. Yeah, first of all, let me see, what is this? This is bacteriophages. Yeah, binding. Yeah, well, let me mention this before I do anything else. Here you can actually decorate the different kinds of bacteriophages with, can't even see what it says here, but also with gold nanoparticles. You can attach them via file groups and do a programmable assembly of nano architectures using the genetically engineered viruses. And that is what I'm going to show you now a little bit. This is how you do. I don't know why I have. Let me just, I'm sorry about this, but I cannot see a part of my slide here. But anyway, this is a way, again, this is also nothing that I would like to go into so much. What you do here, this scheme shows is basically how you insert a part of DNA into the DNA of the bacteriophage, which then in the infection process will lead to expression of the protein that you want. Gene cloning, as you can see here, is a process by which large quantities of a specific desired gene or section of DNA may be cloned or copied once into the desired, once the desired DNA has been isolated. So let me, so this is the recipe for this. You do bacterial cultivation, plasmid purification. Plasmid is nothing else but a DNA. I don't want to dig in too much into the molecular biology here, but what you do is that you insert DNA. Here it is called a plasmid. You can cut different kinds of sites here in this plasmid in order to insert what you can see here in the very middle, GCN4. GCN4 is a highly interesting gene which codes for a protein which is called, yes, hang on a sec. I will skip those things. This is too much. Sorry about that, there's too much molecular biology. What you can see here is the GCN4 has been, when you clone this gene, when you put inside this gene and then you replicate the viruses inside the bacteriophages inside a bacterial cell, that means that more bacteriophages will be produced, including this protein. And why is this, as you see here at the end, right? Why is this protein interesting? It's interesting because you can change it. You can make it more and more specific. You can make it specific in the sense that some bacteriophages connect to each other via this protein and others do not. That means you can do a systematical buildup of nanomaterials. And oh yeah, I skipped this. This is here we are talking about leucines, different kinds of proteins that you can express and their sequences. I will, this is a sequence alignment of the proteins that we have. Those are the amino acids. Excuse me, those are the base pairing. I will save you from all those things. I can mention so much. The GCN4 belongs to the coiled coils. That means a particular protein that is coiled. But those coils, here we have two coils, they are coiled also into each other, which makes their specificity so much higher, and that you can also, here we have an example of this. These are two bacteriophages. Those are TEM pictures that bind because of this GCN4 dimers. You can get longer chains out of this. You can make fibers out of this simply by using this very specific cloning and this very specific GCN4 protein. Here again, this is one of the best results. It's a tripod made out of bacteriophages, which has also been modified in such a way, also with the GCN4 protein. So again, this might not be very spectacular structures. As you can see, we are far away from the Eiffel Tower, but still it's a step in the right direction. Here again, we can do a lot of those things. We can assemble GCN4 peptides by increasing and decreasing the pH and so there are many switches, many different kinds of tools that you can use in this particular field. Again, as I said at the beginning, this is a little bit of basic research, but it's interesting to see how we can build materials at this level. I mentioned already in the beginning, we are talking about bottom up. I don't think I have to explain this. Bottom up and top down, you all know this. But top down, you start with an iron ore and you hammer it, and out comes a wrench. If I simplify this very much, the bottom up fabrication is the other way around. You start with surfactants and molecules and some ions, and then you shake it and you change the pH and the precipitate the wrench. Well, we all know that this is not exactly true, but it is showing the principle of bottom-up fabrication. Maybe one day we can precipitate a wrench, actually, who knows. DNA architecture, promised that I wanted to tell you a little bit about other things. DNA can be used, since it's very complementary material. You can, I also go through this quite quickly, I think. Here you can, by doing DNA origami, as it is called, you can use the sticky ends of the single-stranded DNA and make even a three-dimensional sort of coupling. Genau, exactly. Base complementary. Smileys has been done, stars has been done in this way. What you do is you fold a long single-stranded DNA, excuse me, with so-called staple strands here, and you bind those location together accordingly. So this is sort of a gallery of different kinds of structures that you have that has been made. A big guy in this field is Dr. Rothemund in New York, I think. 3D DNA architecture can also be done by so-called holiday junctions, where you have four DNAs that gather together in the middle, or almost like a porous nanopore here in the middle. It is actually simple. It depends on the base complementary things, and what has been the complementary basis. That's what I wanted to say. Even a box has been made out of DNA that is more than impressive. You can see the box here in the smaller TEM pictures here. A DNA smiley, as I said. Now I have to point out one thing at this point of time also, and that is at the same time a reason why I am interested rather in viruses than in DNA. DNA has a drawback. DNA as a biomolecular construction material, it is not long-term stable. And you remember what I said about the high temperatures that you can use when you do Archaea viruses, etc. This is not the case with DNA. DNA as a material, construction material, is actually not particularly stable. We also have other examples. This is a very virus which is called T4. It is spectacular because it looks like almost like a spaceship. Also with this, you can design materials. Attempts has been made to make materials out of the feet that are, I just call it feet. It's not a living creature. But this is again a natural. We also have viruses coming from the plant field. Tobacco mosaic viruses look like this, and they are very, very rigid, and they can form small carpets like this, as well as centimeter-long nanowires by cross-linking. If you so wish, you don't really need that, but you can cross-link with gold and silver. And you can do a sort of doctor blade with a suspension out of tobacco mosaic viruses. What else? I mentioned appendices in the beginning. This is a very interesting appendix. I think it is a part that is a part of this is a supramolecular structure which is called HAMI, which is a part of the cell surface. It extends from the cell surface of certain Archaea actually, and it's built up like this. You can see the SEM pictures here, and it is a nanoscopic hook that can be used also to drag nanoscopic materials. I don't have any other better idea about how to drag or even do a sort of tribology actually on the biomolecular nanoscale rather than this particular HAMI. So that's what I wanted to tell you about. I hope I have been giving you a little bit of an outlook into the Biosphere, and I also hope that you maybe got some kind of insights into this. And I also hope that I convinced you a little bit that building up nanomaterials is an interesting part of material science, and an even more interesting part is to build up those materials in the Biosphere. Nanomaterials from and in the Biosphere. Thank you very much for your kind attention. Thank you.
I thank the organizers for giving the opportunity to share my work, and also I'm very much thankful for the participants also, invitees and professors. My title of the topic is related to foams, that is when you have heat exchangers and when we introduce metal foams. And this again, in particular, in metal foams, I am taking porous metal foams. Okay. The practical applications of this type of work we can find in natural gas and solar energy systems. Who are working in heat exchangers and also in heat sinks, thermoelectric generators, automotives. And some of the experiments were also carried out by references like WAFI and PIM, where they have taken the porous fins in terms of porosity and pore density on a plate fin heat exchanger. Performance was experimentally studied. Our motto of this study is using foam metal. What is the enhancement in the heat exchangers? So, and also we are in particular about the PPIs. PPIs here is nothing but the pore per inch. Changing the value of PPI, what is the best PPI value in order to have optimal heat exchangers? So I just move on to my topic. Here we have the compact heat exchanger under investigation. Normally in the literature we find one cylinder or one cylinder which was already worked in the earlier case of our research. Now we are improving it by introducing more number of metal foams. In the geometry of the metal foam is a cylinder, and we have included 5 cylinders. This is here we are also going to fix the values of what is the radius, what is the diameter, and what is the particular values for inlet and outlet velocities. So here in related to porous foams, I am particularly using the REV, that means for working in problems of continuity of porous materials. Professor Wafai has given in his textbook in detail about the REV techniques. So I'm using that REV technique. And also LTNE model. LTNE model is local thermal equilibrium is LTE, and LTNE means local thermal non-equilibrium. What is the meaning of this one is, here we have a fluid and we are introducing a porous foam. So porous foam is a solid material. So when we are using a porous foam, a solid material, so you have two different types of materials. One is the fluid and another one is the solid. So when we go for the temperature, when we fix the foam inside the fluid, you will have, you know very well that the conductivity of the fluid and conductivity of the solid materials are different. So we have two different approaches here. We have LTE. LTE means the local thermal equilibrium. That means the fluid and the solid, they will have equal temperature. LTNE means they will have a different temperature. So if you go in detail about LTE and LTNE, it will tell under what circumstances we can adopt LTE, under what circumstances we cannot adopt LTE. So it depends. Who are interested, you can just search about LTE and LTNE. In my particular case, I'm considering LTNE. LTNE means the conductivity, the thermal conductivity of the fluid and thermal conductivity of the solid are different. And also because I'm using porous. Actually, mathematically, if you consider in epsilon to be 1, we go back non-porous. So in this particular case, I'm using the porous foam, and when we are using the porosity, so the Navier-Stokes equations we have to introduce the permeability, what is the permeability of the solid, and get the Navier-Stokes equations, which is a common people who are working in this type of works. The derivation, OFI himself has given a very beautiful derivation from Navier-Stokes equations into, for the Navier-Stokes equation, when you have a porous material. Not only that, I'm also including drag coefficient. When we are using porous material, we have three different types of models. One is Darcy model, another one is Darcy-Brinkmann model, and another model is Darcy-Forchheimer-Brinkmann model. So here, when we are using the Darcy model, we have to assume, depending on the type of the flow we are under study, we have to use respectively the different models. So again, in detail, I will not go. You can read some of the textbooks. For this particular case, I am using Darcy-Forchheimer-Brinkmann model. Regarding the models, you can just have a detail, because again, it takes another 15 to 20 minutes to define each model and why we are considering this. Because I am taking this model. So these are the governing equations, which is very well known for fluid mechanics people. Consistency equation, X momentum equation and Y momentum equation. Here epsilon represents for your porosity. So if you take epsilon equals to 1, mathematically, if I substitute epsilon equals to 1, it reduces to non-porous material. So it is a more generalized Navier-Stokes equation. So here, this is your convective term. Then rho P by rho X is your pressure, viscous, and mu F by K represents for your K is your permeability, and CF represents for your Forchheimer drag. Similar equation along Y momentum equation. As I already mentioned, UV are the velocity components. Epsilon, rho F and mu F are the porosity, fluid density and viscosity. K and CF are the permeability and inertial coefficient. Now we have to go for, because as I already mentioned that we are considering LTNE model, so I have to consider different energy equations, that is for fluid and for solid. So this is for your fluid equation, fluid phase, and this is for your solid phase. So now the normal, when we consider any fluid mechanical problems theoretically, what we are going to do in the normal procedure is we non-dimensionalize get some of the dimensionless parameters such as Grashev number, Reynolds number, etc. and we analyze. But here what I am going to do is, I'm not going to do all those things. I fix the scalings here. I fix the scalings. That means what is my width, what is my height, only for, I directly give the physical properties values and go for the numerical solutions using ANSYS print. So what are the values we want here? So in order to have the solutions for this, what are the unknowns rather than the physical properties? For this, I'm using air as my fluid and aluminum foil as my solid. So I know what is the conductivity, what are the properties of air and what are the properties of aluminum foil. So I directly use these values. Regarding the permeability, there are many different correlations. In particular, I have taken the correlation from Kalmadi. Of course, you can use different types of correlations, but why particularly I have taken this correlation is, Kalmadi has done experiments and he found that this correlation is best fitted. Similarly for drag coefficient. And also from Kalmadi and Mahajan, I am taking this correlation for diffusivity and HSF, alpha HSF and HSF. Again in HSF, depending on the value of a local Reynolds number, I use different relations. If I take Reynolds number less than 40, I use the first. If I take between 40 and, you know very well, Reynolds number, depending on the value of Reynolds number, we can judge whether the flow is laminar, turbulent, so those things. So instead of going for the usual normal procedure of solving Navier-Stokes situations, what we are going in this phase change materials, what we are going to do is, we particularly fix the material and directly put the values of these physical parameters. There is no, of course, these solutions are valid only for this type of materials. You cannot generalize it like other papers. So here we define our Reynolds number, Prandtl number, which are well known. And now, yes, I have to fix the values of the height and the width. In figure 1, here H total refers for the length and LMF refers for the width of the duct. What are the values? So here I again substitute the half of the pitch. Pitch here is nothing but the distance between one cylinder to other cylinder. We have taken 5 cylinders. Pitch represents the length between one cylinder to other cylinder, and the height is LMF. Here for this particular problem, I have chosen L1, L2 to be 0.2 and 0.8 meters respectively. And also some of the things which I am going to fix is about T by L, where T represents for your foam thickness. I am changing the thickness. I can change the thickness of the foam by assigning different values for the ratios T by I. Those things I will explain when we consider the when we run the program. So before that, this work has been computed using ANSYS Fluent 15 code, and while running, who are using ANSYS Fluent, you know what are the options we have. For this particular problem, I have used a simple algorithm, and in order to calculate the pressure, I have used the Presto algorithm. And for the convergency criteria, I have taken 10 raised minus 5 for continuity, 10 raised minus 8 for energy equation. And further, in order to justify for the convergence, we normally do the grid analysis. So this part also I have done. Fixing the T by I to be 3 by 4 and inlet air velocity to be 0.511. These are all about more of mathematics, but this is essential to understand also. So with Reynolds number 392. And in order to do the grid analysis, what I have taken 3 meshes. First mesh with 28,500. Second one is 1,14,000, and third one is 4,56,000. So when we do the grid analysis, the error and the percentage of error between 1,14,000 and 4,56,000 is only 0.3%. So I choose 1,14,000. That is more than sufficient for our convergence. Not only that, we have also done the normal validation with the previous published papers, where we have taken non-porous. So the values, that means if I fix epsilon equals to 1, I get back the results for solid without porosity, which is given in reference 6, and we got, these are all about the computational. For really working, they know in detail. And the value of Reynolds number 56 to 1,120, the range of Reynolds number which has been used. And when we are using the fluent, you know, we have to go for these values only. That is reference values is 288.16. This value we derive, you know, with the help of Gresham number, we calculate the inlet temperature, which is 288.16, and wall temperature is 353, and physical properties are evaluated with standard value, that is 300 K. Okay, this is your mesh cross section. We can visualize very well when you use Gambit software. Anyhow, I have zoomed it. This is how it looks when we do the grid generation or meshing. So geometrical mesh foam configuration, because we are using the cylindrical foam, naturally the curvature will be circular. This is your T and this is your capital I. So I am going to take the ratio, thickness and length. So depending on the value of thickness, which is a very important contribution, how much thick we have to consider the foam in order to have the optimum heat exchange is very important. So this is this paper is already present, published in ASME heat transfer. So if you just click my Google Scholar, you will get my ResearchGate or Google Scholar. You can download this paper for more information regarding the values. Now we will go for what happens, and particularly here we are using the LTA. So pressure fields for Reynolds 392 are reported for different values of feedback height. And in this figure, you can observe that in figure 5, you can observe how the pressure distributes when we consider the ratio thickness and length ratio to be 1/4, half, 3/4, and 1. The temperature field for fluid. These are all about the results we have drawn. Next, I will go for the conclusion part. So we have carried out the numerical investigation on heat exchanger with aluminum foam characterized by porosity. Why particularly I have used aluminum foam is, you know very well that of course we can go for different metals, but aluminum is more of flexible nature and it is also very cheap. So when you adopt aluminum foam in any of the machines, the cost will not be more. That is also one of the important reasons for using aluminum foam. So characterized by ppm, that is, I have already told the 4 per inch. So if I consider ppm to be 0.95 and 20 has been accomplished in order to estimate the thermal and fluid dynamic performance of heat exchange system, we found that ppm 20 is the best value. So keeping that as the equation, we have carried out different thickness for the foam. So the results in terms of heat transfer ratio, higher thickness improve the heat transfer respect to the case with a lower quantity of the aluminum foam. Even the greater value of P/I, the increased porous thickness causes an increment in terms of pressure drop. So if you increase the thickness, the pressure will be dropped. So for oxygen, the ratio Q over W, that means Q represents for your volumetric flow, is large for system with smaller values of metal form thickness, demonstrating that this thing is heat exchanger is characterized by it. So this type of with PI 20 and with this value of P by I, we get a better performance. So regarding the temperature switch also, it is possible to say that aluminum foam, the fluid phase, presents a temperature difference higher than the one in the solid matrix. Solid matrix means without porous. So there also we have used aluminum foam, but without holes in the mesh, without holes in the aluminum foil. So if you use holes in the aluminum foil, we get a better performance of the foam. That's the references, and I have already told who wants to go in detail about this paper. Available online. It's completely recently transferred session this conference. I thank organizers and participants for patience here. So, Navas?
So, am I audible?
Yes.
Thank you very much. I thank the organizers for this opportunity, and I express my greetings to all the speakers of this session. And we heard wonderful talk from Professor Lothman on the latest topic. And I'm going to continue my presentation on a piece of work, what we have done to take care of nuclear waste. And the topic of this talk is conversion of rare earths of hazardous nuclear waste into somewhat useful potential material based on apatites. So as we all know, we continuously develop technology to fulfill our needs. At the same time, all of us know we need to take care of the side effects in any form. And it's so clear that nuclear reactors help us generating energy. At the same time, there is parallel generation of waste. So we need to give equal importance to take care of this waste. And we have done a small piece of work towards this end. And all of us know the process which involves in the conversion of nuclear fission, the energy which is generated during nuclear fission into the required form of energy, that is electrical energy, as it is represented in this pictorial way. And all of us know this nuclear power generation is an alternative approach, alternative source, mainly from the greenhouse effect. And we are in the safer, almost in a safe zone as far as the emissions are concerned. At the same time, we are not in a safe zone as far as the taking care of, or as far as the waste which gets generated during this process. And the entire process of this nuclear power reactor generates different levels of waste based on their activity, radioactivity level. We come across low level, medium level, there is another intermediate and then high level. Our focus is on taking care of this high level nuclear waste. This is a general composition. The composition varies with respect to the type of fuel being used, with respect to the type of reactor under use. So generally, the composition of the waste contains the various elements of our periodic table, and mostly actinides present in it. And our focus is to take care of the rare earths present in this nuclear waste. And what are we doing currently regarding taking care of this or disposal of this high-level waste? And eventually, after doing some process, we go for deep geological disposal. And it's so clear that directly we cannot go ahead and dump it. It's impossible. So we need to do a process in between. What is that process? We need to contain this waste in an inert host or a matrix, and then we need to keep them in a layer of insulating. Insulating in a sense, thermally, radioactively insulating layers of materials, and keep it in a canister, and then only we can go ahead and go for geological disposal. So one of the processes is this containment of this waste into a solid form, that is referred to be waste form. And it has to be thermally, chemically, and also radioactivity resistive, and thermally and chemically the host or the embodiment has to be stable. And over a period of time, globally, people have been working on various materials, right from cement. And as far as the materials are concerned, I just bring over here the given types. Glass matrices, glass materials as hosts have been extensively studied. And then comes ceramic hosts. And as far as glass hosts are concerned, you can go for loading very relatively high percentage of load of waste. At the same time, we can imagine geological burial means there is an environment which has high temperature and flow of liquid. All these things are there. So, and hence there is a concern regarding de-vitrification of this glass, and which contains this material, that means radioactive waste. And if there is de-vitrification, you can very well expect the leaching of species which have been contained in this host cannot be, or it cannot be avoided. So once it gets leached, then you can expect contamination. So that's a trouble associated with this, but lot of work are going on. And then another host is ceramic-based matrix, and people have been working on different types of ceramics, and even a combination of glass with ceramic also people have worked. And synrock, which is a combination of nearly 4 ceramic materials like rutile, hollandite, zirconite, all these types of materials combined to put together, processed, and then have been converted into this type of waste form. Wonderful. Because of the presence of 4 different matrices, you are in a position to load a high percentage of waste. At the same time, because of the same presence of 4 different structures, they are structurally, chemically, they are different. With the condition of the geological burial site, there is always a chance for the mismatch among them when they try to respond, when they respond to the heat and then flow of the fluid. And hence, there is another matrix, which is a single phase. It's not multi-phase as in the case of synthetic rock. It's a structurally single material we identify as a host, and then try to immobilize the species present in the waste as much as possible. Since it is a single phase, the challenge here is loading higher percentage of waste. Otherwise, the differential thermal response and then structural integrity problem, whatever one can expect in synrock, that means multi-phase materials, cannot be expected in this type of single phase. Our focus is on this single phase crystalline materials. So in our lab, we have developed the waste forms based on these different types of inorganic materials. They are hosts. One is based on sodium zirconium phosphate, another one is based on monazite. And then we worked on lanminite, and also we have done some work on apatite. And today I'm going to discuss the work we have done on apatites. And based on these 4 types of framework inorganic materials, we have developed this type of multi-ionic based forms containing different ions in sodium zirconium phosphate, monazite, and then lanminite and apatites. Under apatite, we have developed based on oxysilicates and oxyphosphate. And today I'm going to focus on oxysilicate. Apatites are wonderful naturally. They are existing naturally, available naturally as a mineral. It's a structurally very interesting material. It is formed by 2 types of chains. The general formula contains Ca 5 times, PO4 thrice, X can be hydroxide ion, fluoride ion, chloride ion, bromide ion, oxide ion. All these possibilities are there, and generally I represent that as X. So the framework is made up of interconnection between 2 types of chains. One chain is made by interconnected CaO9 polyhedra. Another chain is made by CaO6 interconnected chain. And these chains are further built together by PO4 tetrahedra. So there is a chance because of the presence of these ions in different crystallographical sites, it is possible to introduce various ions. That is the versatility of this structure, which crystallizes under hexagonal system, and the space group is indicated here. And why did we choose this type of materials as a host? Because the literature report indicates very good thermal stability of this type of materials coming under this apatite category. Similarly, they exhibit good chemical stability. And in addition to this, even though we have not done the radioactive resistive study, but literature report exhibits very good radioactive resistance of these materials. And that's the reason behind choosing this type of host. The parent oxyapatite we have chosen has the formula CaLa4 thrice O. There are 2 different crystallographic sites, as I have mentioned before, 9-fold 4-up site, 6-fold 6-up site. In this parent, the 6-fold site is completely occupied by La3+. The 9-fold site is shared by both Ca2+ and La3+. This is the distribution of the ions in the skeleton formed by silicate. What did we do? First step, we wanted to know what is the optimum level we can introduce various rarents. We have chosen lanthanides from safety point of view, and we all know lanthanides mimic or actinides mimic the property as that of lanthanides, and hence we have gone for this lanthanides. And various lanthanides, lanthanide ions we have done, and with the different range also we have carried out. And also we wanted to know only with the rare earth substitution what happens. And in addition to rare earths, if we bring some alkali metals, what happens? And hence after the optimization, we prepared 2 types of complex composition. One we refer it as SWF1, which contains only rare earths. Second one, along with the rare earths, we have introduced lead and strontium. We refer this as SWF2. And basic characterization, we prepared these materials, these compositions by solid state method, by grinding together all the oxides and heating them to very high temperature at around 1200 degrees. And then we wanted to know the pure phase formation. So we have carried out the characterization by powder X-ray diffraction method and then infrared spectroscopic methods. These are the features of apatite structure, and we noticed they are pure, number one. Number two, we also noticed the slight change which happens in the unit cell with respect to the various ionic substitution, and which is indicated here by the calculation of the unit cell dimensions. Since it is a hexagonal unit cell, we have A and C lattice parameters. And when you compare the lattice parameters or the unit cell size of the two waste forms we prepared with the parent, only with rare earths, we notice there is a shrink in the unit cell, shrinkage in the unit cell. On the other hand, the unit cell shows expansion when we have, along with the rare earths, when we have this lead and strontium. It is obvious to know the reason. The expansion with the presence of lead and strontium is due to their larger size compared to calcium. On the other hand, the smaller size of the unit cell after introducing the rare earths is because they are all the substituted rare earths are smaller size compared to lanthanum 3+, and hence there is a shrinkage. Now, we wanted to know how far, because we processed this at very high temperature for the preparation, how far we can expect the presence of these ions with respect to theoretically calculated quantity. And interestingly, we noticed in most of the cases, except lead, in most of the cases, we noticed there is comparable quantity in terms of weight percentage of the ions we have introduced. The analysis is, we call it as EDAX, elemental analysis. And then basically, in order to propose the possibility of using these compositions as waste form, we studied two properties among the three properties required. One is thermal stability, another one is chemical stability. And thermal stability, the first one, we carried out a thermogravimetric analysis, which shows compared to the parent, the waste forms are better. They don't undergo any degradation. And the mass loss is, or the weight loss is very, very feeble. Then, okay, the bulk response with respect to heating is like this in this temperature range. What might have happened, unit cell size with respect to the unit cell. So we have carried out high temperature X-ray diffraction studies to understand the behavior of the unit cell, which is the fundamental unit of any given compound, which is a thermal expansion. And we carried out thermal expansion coefficient based on our data. And in general, we can categorize low, intermediate, and high thermal expansion materials based on the thermal expansion coefficient. And we wanted to study among these three categories, under which category our materials are going to be. And the systematic high temperature thermal studies based on high temperature X-ray diffraction shows we can very well plot the variation of the unit cell dimensions with respect to temperature. This is first plots indicate the behavior for the parent. Second one, the behavior for waste form 1. Third, the behavior for the waste form 2. So now what happened in all the three cases, when you heat the unit cell along the three dimensions, A becomes equal to B because it is hexagonal. So there is a progressive expansion along the dimensions of the unit cell. This is general observation. Then we wanted to know what about the coefficient of expansion? Is it going to, even though it shows a progressive increase, whether the coefficient is going to be the same or different. So then we did the calculation of thermal expansion coefficient, and we plotted the coefficient variation against temperature. And we noticed in the case of parent, the extent of thermal expansion coefficient along the A direction of the unit cell shows the expansion, which is slightly higher than the expansion along C dimension or C direction of the unit cell. And the calculated average thermal expansion coefficient is given over here. What about the waste forms now? So when we go for the rarest alone substituted waste form 1, the behavior is similar to the parent. At the same time, the thermal expansion coefficient is relatively lower, which is reflected in this lower average thermal expansion coefficient compared to the parent. Now, when we move on to the third one, which is the waste form 2, where along with the rarest, we have this lead and strontium in their +2. Both of them are in their +2 oxidation state, and the behavior is different. So what is the difference? So I can explain. So when we observe the variation of the thermal expansion, for example, let's see alpha C, that is the thermal expansion coefficient along C direction of the unit cell, then automatically we can understand the variation of alpha A. So now what happened, there is a cross point with respect to alpha A and alpha C happens at a particular temperature that is around 673 Kelvin. Something should have been happened in the unit cell at this temperature. So based on our understanding, we had come across a perception, that is, we have this lead and the composition is multi-ionic, which we prepared at high temperature heating. So there is always volatilization of certain percentage of lead, and which might have created vacancy in that particular crystallographic site. So in the structure, this lead present in I can say the 4up site, which decides any change which happens in the C direction. So the formation of this vacancy leads to this change at this particular temperature, because after that there is an expansion compared to the expansion along C direction. So now what happens with the after 673 Kelvin, the change, the alpha A becomes higher than alpha C. That is another perception we made, because the temperature is high, it is going on increasing. So there is, with this thermal input, there is a chance for movement of some of the lead ions or shifting of some of the lead ions from another site, that is 6H site, to this 4F site, and which creates a vacancy in the 6H site. Any change happens in the 6H site influences the property along A axis, and hence after 673 Kelvin, due to the formation of the vacancy, an increase in alpha A occurs. So this is what we have proposed as our understanding. So now overall, the property is anisotropic because it is different with respect to the crystallographic dimensions. And the parent and the first waste form behave similarly, where throughout the temperature range we studied, alpha A is greater than alpha C. On the other hand, the waste form 2 exhibits different behavior because of the presence of this lead. And before 673 Kelvin, alpha A is less than alpha C. After 673 Kelvin, alpha A becomes greater than alpha C. Overall, the waste form, the alpha average of waste form 1 is lesser compared to the parent, and then comes the waste form 2. So the unit cell variation is happening, And I mentioned here the anisotropy we noticed. And we also indicated parent and the first waste form behave similarly. So when we draw the variation of alpha A with respect to alpha C at each temperature, we notice that the preferred axis of the deformation which is happening is along A in both C, the parent and the first waste form. So from this, each circle indicate each temperature. On the other hand, the different behavior which we observed with respect to the waste form 2, which contains lead and strontium along with the rare earths, initially, with respect to the temperature variation, initially the deformation is more along, it is the priority is along C-axis, and when you go ahead and heat, the ellipse becomes different, and the deformation is more along A-axis. So the thermal behavior with respect to the presence of the different ions occupying different crystallographic sites are very nicely explained here. So now, as a material, we noticed our oxysilicates become closer with respect to the property of other oxysilicates in terms of thermal expansion reported. And overall, so far reported materials, when you notice, the current waste form 1 shows the lowest thermal expansion coefficient. So this is what given here as a summary. And the waste forms exhibit a minimal weight loss, and they show thermal stability throughout the temperature range we studied. And the waste form 1, which contains only the rare earths, shows the lowest. And this bar diagram gives an idea with respect to each temperature. Throughout the temperature range, if you notice, the waste form 1 exhibit the lowest value, and hence, even from this, we can propose the waste form 1 could be one of the best waste form. Is it sufficient? No. We also have to go ahead for studying chemical stability. Then we made a monolith out of this. We have chosen these materials, all the 3 materials, and made them into monolith and introduced them into a flow of liquid. Here the liquid flow we have taken in our laboratory scale experiment is double distilled water, which got heated, boiled, and the vapor condensed and introduced into this material, which is kept in the path of it. And then over a period of 40 days, we have carried out. After each day, we have sampled the material and characterized by inductive for elemental presence by inductively coupled plasma analysis combined with the MS combined with the OES. And then we calculated the leach rate based on this formula. And what is the result? All the ions are going out, coming out at this temperature of around 100 degrees C, or anything is left behind. That was our eager. We noticed in all the 3 cases, the rare earths are sitting there very sturdily. They have not come out. Then what about the Si, which is present in all the cases? They show relatively low leaching. And what happens to calcium present in all the cases? The leaching of this calcium is also relatively low. And in the waste form 2, strontium leaches out a relatively low level. For comparison, we have carried out comparison with the related materials. And the behavior, the leach rate behavior, initially it used to be high. Over a period of time, it comes down in all the cases, in all the elements, whatever we have studied. And then what about the phase purity? Is it pure? After the leaching studies, we carried out the X-ray diffraction studies to understand is there any degradation happen? Because we have introduced the material to hot flow, and hence we wanted to know In the case of waste form 1, I present here for waste form 1, which is and for waste form 2. In the case of waste form 1, it is stable. We have not noticed any other secondary phase, and the textural appearance is also almost similar. We have not noticed any other formation. In the case of waste form 2, and because of some surface wash off, the removal of some particles present in the grains, etc., happening during the flow, and very nice. The hump and all have gone out, and very nice pattern we obtained after the leaching studies. There is a small, less intense appearance of a peak, which indicates formation of some lead silicate. And the textural appearance, we notice the smaller particles come together and they are becoming bigger. So that's the change we have noticed. And how it is happening, whether all these ions are coming out just by wash off, or they follow any mechanism. So we can expect a diffusion controlled or dissolution controlled or surface controlled. In order to find out, we feed the data with this expression containing 3 coefficients. Among them, A0 talks about the dissolution, surface was off by dissolution. A1 tells about the diffusion. A2 tells about the complete matrix dissolution. And the data we compiled here for waste form 1 and waste form 2, only for the leached out ions. And in the first case, both the ions, calcium and the Si, they follow diffusion controlled leaching, because A1 is higher than A0 and A2. And then in the case of waste form 2, the calcium follows dissolution mechanism, strontium follows diffusion mechanism, and silicon follows dissolution mechanism. So based on our thermal and chemical stability studies, we do propose the waste form 1 of this oxysilicate containing rare earths exhibit good property, and hence for the immobilization of rare earths present in the high level nuclear waste, one can very well go for containing in this oxysilicate matrix. So this is the finding we observed in our study. And I'd like to thank the various agencies which helped us to develop, fabricate the materials, go ahead and do a lot of studies and come up with these findings. And I thank our students who are passed out students, Ms. Ramya and Mr. Pradeep Kumar, for their wonderful work in this regard. I thank you all present here for your patient hearing. Thank you once again.
For the invitation to this conference. Today, I will do a short review related to sputtered bioactive thin films. These thin films were prepared in our department. So I'm Alina Vladescu from National Institute for Optoelectronics. I'm working at the Department for Advanced Surface Processing and Analysis by Vacuum Technologies, which has the main topic, the preparation of thin films by different PVD methods, such as magnetron sputtering, arc cathodic, and so on. And also we have possibility to do some plasma surface treatments, and also we have experience with system, ultra-high vacuum system. Related to the coatings, in our department, we have different application of coatings. So we can prepare hard coatings, as I presented here on different systems. Also, we have possibility to obtain decorative coatings on plastic, ceramics, and glasses substrates. In the last years, we also prepare the coatings with high reflection coefficient in ranging in different ultra visible domain. And not the last, but we also start to work with biocompatible coatings used in different field of medicine, something like implants, prostheses, and also the medical instrumentations. My talk will follow the 5 steps. So I will start with a short introduction. I will present the technological challenges of my talk and experimental details and also the results and discussion. So why is a biomaterials? As you know, biomaterials exhibited many advantages, some such non-toxic, which means biocompatibility. Also, in some cases, it is needed to use biomaterials without pores. Also, it's important to be stable and mechanical strong enough to withstand the repeated forces during the lifetime of implants. Also, the biomaterials, for biomaterials, it's important to work in environment of our body, which is not a constant environment, because we have a different temperature which can be varied. Also, we have a corrosive environment due to the salty chloride solution of our body, and also in various places of our body we have different pH values. So we have an unstable pH in our body, and also in different places of body we can have different mechanical stress. So in this present, there are a lot of biomaterials used in real life. Many of these are based on the polymeric materials, and also there are biomaterials, metallic biomaterials used in medicine in various fields, as I presented here. Unfortunately, most of the biomaterials, whatever is substrates, polymeric, ceramics, or metallic, have major complications. These complications are related to the aggressive environment of our body, which is corrosive, highly corrosive environment, and this environment will lead to a highly corrosive process, and most of this process will destroy or will degrade the implants. Also, the second important process in our body is wear. In most of the cases, wear processes will also lead to degradation of implants due to the release of particles into our body. Also, despite of aggressive environment in human body, we have a different bacteria, and most of the implants prove to have low antibacterial properties. And in some cases related to the metallic implants, these metallic implants have a low osteointegration. So this, all these main complication will limit the application of metallic material as an implant. So in general, in order to increase the corrosion and wear of metallic implants, was developed a novel material, and also by using different processing techniques in order to increase the biological interfaces between metallic implants and human tissues. So this here I presented just two examples to increase the process to increase the corrosion and wear property: oxidation treatment and also the coating-based anti-corrosive and hard coatings. In order to increase the antibacterial properties of metallic implants, here I presented just two methods. So coatings based on silver and copper, and also I found some peptides layer. For increase the osteointegration of metallic implants, the most used method is to prepare the calcium phosphate based coatings on metallic implants. So in our department, we try to increase the properties of metallic implants by different solutions. So one of these is to coat the metallic implants with coatings based hydroxyapatite. Usually the hydroxyapatite is very good. It has a good osteoconductivity, but this has some disadvantages, such low corrosion resistance, low mechanical properties, and also the low antibacterial properties. So in order to increase these characteristics, we tried to dope hydroxyapatite with different small amount of silicon carbide, silver, and titanium oxide in order to increase all these properties of hydroxyapatite. As I mentioned, why hydroxyapatite? Because you see here, this hydroxyapatite has a lot of disadvantages, but despite of these disadvantages, hydroxyapatite has a huge advantages which are related to the high ability to integrate in our bones, and also can support the ingrowth of new bones. So as I mentioned, we try to increase properties of hydroxyapatite by silicon carbide. Why silicon carbide? Because this type of coating exhibited a good resistance to corrosion in corrosive environment, has a low dissolution rate, and also have enough mechanical properties compared with hydroxyapatite. And also, in some cases, it's inert, it's chemically inert material. For increasing the antibacterial properties, we add some small amount of silver into the hydroxyapatite, and to improve the relation between tissue and metallic implants, we coat the implants with titanium oxide. The method used for deposition was the magnetron scattering in RF regime, and here you can see the RF power feed on each target used for coating. As you see here, I will present just one example for each one because this condition proved to have the best anti-corrosive properties, antibacterial properties, and mechanical properties. Until to obtain this condition, we prepare for each coating, we prepare a lot of different parameters and investigated all of them. We kept the deposition temperature at 700 degrees because, as you know, probably the hydroxyapatite is crystalline when the substrate, when a deposition temperature are more than 500 or 600 degrees. Regarding the results, as you see by EDS, we show that whatever was the addition dopants, the hydroxyapatite structure is still there, and the ratio between calcium and phosphor, it's more or less close to the stoichiometric hydroxyapatite. So this is an important result because the addition of dopants doesn't affect the formation of crystalline of the calcium phosphate phases. About the SM analysis, you can see that all coatings exhibited a smooth surface without cracks or scratches or other defects. Here are presented the XRD diffractograms, and as I said, the hydroxyapatite undoped, it's crystalline, has a crystalline structure. Here you can see the hydroxyapatite, the peaks attributed to hydroxyapatite. So by adding the silver, it still obtains a crystalline structure. But after the addition of silicon carbide as well as titanium oxide, it can be seen that the hydroxyapatite will be a coating with amorphous structure. It was not unexpected results because it is well known that the titanium oxides usually obtained by PVD method, it's a amorphous coating. Also, the TR analysis proved us that whatever was the doped element, the hydroxyapatite is still there. Sorry. About the AFM results, we can see that hydroxyapatite without doping elements has a rougher surfaces. By adding the silver, the roughness of the coatings increased, but by adding the silicon carbide and titanium oxide coating dopants, the roughness of hydroxyapatite decreased significantly. Regarding the nano indentation, you can see here that the elastic modulus of the coat of the hydroxyapatite coatings will decrease for adding the silver and silicon carbide, but increases for the element for the doping element of titanium oxide. Related to the harness, all additioning all element, all dopant element increase the hardness of the hydroxyapatite. More evident, it's for the case of silicon carbide, which this hardness probably is increased due to the silicon carbide, which has a high mechanical properties. Regarding the contact angle, you can see that all coatings exhibited a low contact angle compared with uncoated substrates. So all these coatings are more hydrophilic than uncoated substrates. And about addition of doping element, you can see that the doped coatings are more hydrophilic than the that's the undoped coating. In order to analyze the corrosion resistance of all coatings, we try to test the corrosion resistance in 3 different solutions in SBF, PBS, and also the MEM solution. We also keep the human temperature, and what we saw here based on polarization curves, we calculated the main electrochemical parameters. So what we saw that whatever was electrolyte, all coatings improve the corrosion resistance of uncoated alloy. By comparison, developed coatings investigated in SBF, we can tell that the coatings doped with titanium oxide exhibited the best corrosion resistance because exhibited the low current density and high polarization resistance. The same results were obtained in the case of the solution on PBS and the MEM solution. Anyway, it is important that by adding silver and silicon carbide and also the titanium oxide, the corrosion resistance of undoped hydroxyapatite was improved, whatever was the electrolyte. This is an important result. Also, we tried to see which is bioactive ability of each coating. These bioactivity abilities were investigated after 1, 3, 7, 14, and 21 days. By immersion, each coating in 3 solutions, the same solution as we selected for the corrosion resistance. So we keep each coating for this immersion days, and then we investigated the surfaces after each immersion day. So here you can see the coatings after here I presented the evolution of apatite mass evolution. What doesn't mean? we try to see which was weight of each sample after each immersion day. So you can see here in the MEM solution, each coating speeds up the biomineralization character. What means this? The weight of each coating increased. So this means that the biomineralization process was increased. About the immersion of coatings in PBS, you can see that all coatings exhibited degradation effect. So this solution, no, it means that the coatings are not resist to this solution. Regarding to immersion on SBF, we can see in some cases degradation process, and also in some cases biomineralization effect, depending on the type of coating. Here we tried to show the SM and ADS results after each days of day of immersion, and in different corrosive solution. If you see here in PBS, the coatings are still have a ratio between calcium and phosphor close to the hydroxyapatite. But after the immersion in the MEM, you can see that the ratio between calcium and phosphor increased a little bit, which means that the coatings change the structure from the hydroxyapatite in one related to the calcium phosphate phases. And also the same results can be seen in SBF solution. If we compare the SM images, we can see that after, for example, after 21 of immersion days in SBF, part of coatings are delaminated, completely delaminated. Also, we try to see which kind of phase we have of after each immersion day and after immersion day in different solution. So we saw that after 1 and 3 days of immersion, the coatings are stable in all investigated media. After 7 days, the intensity of the substrates is more obvious. This means that the coatings start to be degraded. After 21 of days, we saw that we saw the apparition of new phases based on calcium phosphor phase. Here, we also presented some SM images and also the EDS analysis for coatings doped with silicon carbide in all 3 media. We didn't see a significant difference between the MEM solution and PBS. Also about the XRD, we also saw that the intensity of the peaks on spectrum immersed in PBC solution is lower. This means that the coatings it's almost degraded in this after the immersion in PBS. And here are presented the SM images of the coatings doped with titanium oxide. Also, in this case, we couldn't see the significantly differences between the immersion of coating in all three media. And here I presented the ratio between calcium and phosphor in the case of coatings doped with titanium oxide. So we didn't see the changes during these immersion days. The same results were also obtained by FTR measurements. Also, we tried to see what is antibacterial properties, and these tests were prepared on Staphylococcus aureus bacteria, and we have used 3 different concentrates of media. The tests were performed during 24 hours and also after 7 days. So we found that any coatings exhibited a negative reaction, and we saw some positive reaction related to the uncoated alloy. The same results were obtained also after 7 days, so it was not important the period of the test, because we had obtained the same results, and also the same results were obtained whatever was concentrated or diluted the solution of media. We also investigate the antibacterial properties by using the Salmonella bacteria in the same concentrated and diluted environment after 24 hours and 7 days. So the results are the same as the previous bacteria. So a positive reaction just on the case of uncoated alloy, and a negative reaction for all investigated coatings. As a conclusion, so we can see that the addition of silver and silicon carbide decrease elastic modulus and increase the hardness, and while the titanium oxide increased both elastic modulus and hardness. About the corrosion resistance, we can see that all coatings exhibited a good corrosion in the MEM solution, and a low corrosion resistance in SBF and PBS media. About the degradation or biomineralization processes, all coatings exhibited a good mineralization abilities in the MEM solution and a degradation abilities in SBF and PBS media. All coatings proved to have a good resistance to bacteria, to E. coli and Salmonella after 1 day or 7 days of tests. So we can see that we can say, to summarize, that the hydroxyapatite properties can be improved by addition of small amount of different elements, and we just to select which is the final application of material, and then we can tell which element or which material can be used. So if you see here, the results is not a material is not good for all tests. So in biomedical application, it's important to do a selection. sometimes this means a compromise. It is impossible to obtain a good, a superior biomaterial used or proper, more exactly, proper for all biomedical applications. So in this case, we should do a compromise. What kind of properties is most important for the final application? So firstly, I would like to thank to my group, and also the results are obtained during the national project, and also I would like to thank to some colleagues from University Polytechnica and from Dokuz University from Turkey, and also to Mihaela Bada for the antibacterial properties that I presented. And also, I would like to thank you for your attention. This is our lab.
Let's discuss carbon capture applied to the Portuguese cement industry. I'm Lisa Bacatil. I have a master's in materials engineering, and I'm currently doing a PhD in sustainable energy systems while working in the cement industry with the goal of decarbonizing the sector. The presentation is divided into 5 parts. I'll start by explaining the motivation behind the chosen subject and the cement industry, followed by a brief introduction to my organization, C5 Lab. Afterwards, we will review the concept of carbon capture, in particular its application in the Portuguese cement industry. Finally, I will present you a conclusion and the next steps of the project. Starting with the problem. Environmental related challenges were the driving force for the European Green Deal strategic vision. It aims to achieve net emissions of greenhouse gases by 2050, and its relevance is now reinforced by the COVID-19 pandemic. However, there are industrial emissions from sectors that will be difficult to eliminate. The manufacture of cement is a major contributor to climate change, as it is a CO2, energy, and material-intensive industry. The cement sector alone currently results in a large quantity of around 7% of the global anthropogenic greenhouse gas emissions. So reducing CO2 emissions from the cement industry is becoming a global imperative. On the other hand, the concrete industry, and therefore cement, is essential to the global economy and development aspirations, as it is the most widely used man-made material in the world. And it will be a major constituent of new buildings and infrastructure that will meet the world's housing, business, and transportation needs in the 21st century, as there are no foreseen substitutes of the key attributes of concrete: strength and resilience. This increased demand for cement and concrete is mainly related to the rising global population and urbanization patterns, coupled with infrastructure development needs. To understand the challenge of producing carbon-free cement, we need to understand how cement is made. Its production is divided into 10 main stages, from the pouring of raw materials to the storing in the cement silo. But the most energy and CO2 intensive process is stage number 6, which is the clinker production in the rotary kiln. In the first stage, naturally occurring calcareous deposits such as limestone and marl provide calcium carbonate, which is a key ingredient for cement. They are extracted by heavy-duty machines from quarries, which are often located close to the cement plants. Small amounts of other materials like iron ore, clay, or sand may also be extracted from deposits to provide the extra iron oxide, alumina, and silica needed in the chemical composition of the raw mix to meet the process and product performance requirements. In the second step, the quarried materials are crushed and transported to the cement plant. Afterwards, the raw materials are mixed together in different proportions in a process known as pre-homogenization to create cements with specific chemical compositions. Material then passes through a preheater and precalciner and is finally heated in the kiln. The intense heat triggers the chemical and physical changes that transform the raw feedstock into clinker. During this process, calcium carbonate is gradually heated with small amounts of additives in a kiln. At approximately 900 degrees, calcination occurs and the CO2 is released from the calcium carbonate. With additional heating at around 1550 degrees, the calcium oxide reacts and agglomerates with silica to form clinker. And the most energy-intensive step in the cement production is this process of clinker burning. You see, the cement sector is facing the challenge of meeting an increasing demand for its product while cutting direct CO2 emissions from its production. The problem is that, as I've shown you, the CO2 emissions are intrinsic to the process itself, and around 842 kilograms of CO2 are emitted per clinker ton. To answer this challenge, 5 entities in Portugal gathered as one to form C5 Lab. 2 only cement companies from Portugal, Cecil and Simpor, together with the Cement Association ATIC, the Technical University, and the LENEC Laboratory. The C5 Lab is a Sustainable Constructions Materials Association and R&D platform for the development of innovative technologies for the sustainable production of cement with a reduced CO2 footprint. C5 Lab aims at contributing to a carbon neutral society by focusing on 3 main areas: energy transition and efficiency, sustainable cementitious materials and innovation in manufacturing, and finally, the area I'm focusing on, carbon capture, use and storage, known as CCUS. In fact, to meet the European Commission's expectations, we need breakthrough technologies. According to the European Cement Association, CEMBERO, the integration of emerging and innovative technologies like carbon capture is identified to provide the largest cumulative CO2 emissions reductions by 2050, with 42% contributions. A possible solution to achieve this goal would be to have 59% of the cement plants equipped with carbon capture technology. There are 3 main carbon capture methods: pre-combustion, post-combustion, and oxy-fuel. In pre-combustion, an air separation unit produces a stream of almost pure oxygen, which flows into the gasifier and reacts with fuel to form syngas, which is a mixture of hydrogen, carbon monoxide, CO2, and water. Then, the steam is added to the syngas in the shift reactor to separate hydrogen and the CO2 produced. The CO2 is captured from the gas stream and after dehydration and compression is ready for transport and storage or use. In post-combustion, the flue gases exiting a combustion plant are treated to selectively remove CO2 from the gas mix. This downstream separation of CO2 can be achieved mainly using chemical or physical sorbents. One of the main techniques used involves passing the flue gas through a water-based solution of monoethanolamine, or MEA. The nitrogen and the CO2 released by the process are redirected to the carbon capture unit and cooled down. While the nitrogen is released into the atmosphere, the CO2 reacts and binds with an amine liquid in the absorber. The resulting rich amine is heated up, so the solution splits up again into CO2 and amine liquid. The CO2 obtained is about 99% pure, and it's directed into a compressor to be transported away. Finally, the oxyfuel method. The combustion process uses almost pure oxygen instead of air. An air separation unit removes nitrogen from the air, producing oxygen, which is injected with the fuel into a boiler where combustion takes place. Steam is generated and used to power turbines, transforming it into electricity. The flue gas of carbon dioxide and water vapor is recirculated to control the temperature in the kiln and cooled, leaving the captured CO2 to be compressed and dehydrated. Well, regardless of the carbon capture method we use, after capturing the CO2, one question arises. What do we do with it? So we have two options, known as carbon capture and storage, CCS, or carbon capture and utilization, CCU. CCS technologies separate the CO2 produced by the industry, transform the captured CO2 to specific locations, and then store it permanently in underground deposits, such as deep saline formations, depleted oil or natural gas reserves, deposits in unminable coal beds, and other geological formations. CCU technologies, on the other hand, transform the captured CO2 into high value-added products, such as materials and fuels. CCU is considered as a possible alternative or complement to geological storage of CO2 and can increase the economic value of the captured CO2. However, the implementation of CCUS requires specific in-depth environmental and economical analysis to explore different pathways for its implementation in distinct contexts. Considering that the Portuguese low-carbon roadmap for 2050 identified the adoption of CCUS in this industry as policy for abating greenhouse gas emissions, at C5 Lab, we are focused in defining criteria to assess the decarbonization potential and economics to retrofit Portuguese cement plants with CCUS. The methodology we are using in this project is divided into 5 main steps. First of all, we need to identify the possible locations and specific cement plants to implement this technology. Afterwards, we will review the carbon capture methods that can be used. We then compare the cement decarbonization potential of the options previously selected. This cement decarbonization potential is measured in terms of the annual CO2 avoided and the CO2 capture rate. After that, we will submit the alternatives to an economic evaluation by comparing their payback periods, the net present value, and the internal rate of return. Finally, with the preliminary results, we will reach the carbon capture method recommended for each location at the moment. Out of the 4 locations identified as potential clusters for CCUS, 3 of them include cement plants. Location A, between Lisbon and Setúbal, includes 2 large-scale cement plants, Simporarandra and Cecilotão. Additionally, it's located near a paper and pulp mill and 2 power plants. Location B, in the northern part of the region, includes the Simporçozelas plant, which is somewhat isolated at more than 30 kilometers from the nearest sources, namely 3 paper and pulp mills, 1 power plant, and 1 glass factory. Location C, near Marinha Grande, includes 2 smaller scale cement plants, Cecil Cibrapataias and Cecil Maceira Lige, close to sources in the glass sector and the lime production facility. As you may recall, there are 3 carbon capture methods: pre-combustion, post-combustion, and oxyfuel. However, pre-combustion presents a significant disadvantage as it doesn't capture the CO2 emitted during the clinker burning process, which is responsible for 60% of the total emissions, and therefore it's excluded as a potential solution. This leaves us with 2 methods to capture carbon: post-combustion, and oxy-fuel. Now, to evaluate the cement decarbonization potential, we need to consider both destinies for the CO2, storage and use. In the case of CCUS, we need to study where to store it and the transportation until this location. Is it onshore or offshore? Will it be by pipeline or through ships? When it comes to CCU, we can combine the CO2 captured with green hydrogen in a process called methanation. In the Portuguese hydrogen strategy document, this strategy is known as power to gas. So imagine the cement factory emitting gases that contain CO2. By capturing the CO2, we can convert it into methane. The methane is then fed into the natural gas system to which the factory is connected to. And then we have a circuit that recycles CO2. So while selecting the locations, we need to consider the natural gas network for potential transportation and distribution of both CO2 and hydrogen. In blue, you can see the potential storage units, and the current natural gas pipelines are highlighted here in red. Currently, there are no planned new infrastructures specifically developed for hydrogen transmission and distribution in Portugal between 2022 and 2031, but these may change. Additionally, although the distribution of natural gas is being reinforced, since it's more than 100 kilometers north from location B, only the current natural gas networks are considered as a possible CO2 or green hydrogen infrastructure. If we compare the annual emissions of each location, it's easy to see that location C presents by far the lowest emissions. And for that reason, the cement decarbonization potential, as in the annual emissions we could save by retrofitting a cement plant with the carbon capture technology, would be insignificant compared to the other locations. As such, location C is excluded from the analysis. The total CO2 avoided during the project's lifetime was calculated for the 3 cement plants for both post-combustion and oxy-fuel. Results are similar between both methods, but in the worst case scenario, we can reduce 0.5 megatons of CO2 annually, which is equivalent to decreasing the Portuguese CO2 emissions by 1% out of the 7% share that the cement industry is responsible for. Now, to understand if the implementation of technologies is economically viable in each cement plant, we need to do an economic evaluation. And that means playing around with a few assumptions. We started off with a fixed cement price and discount rate. We've also established a reference scenario without CCUS, but it's not exactly business as usual, as it considers all the other complementary measures available to the cement industry to reduce CO2 emissions, like the reduction of clinker to cement ratio. Additionally, we had to assume the CAPEX and OPEX for each one of the 3 scenarios. So without CCUS, with post-combustion, and with oxy-fuel carbon capture. We started the analysis with an increasing price for each CO2 ton emitted through the years, up until 2055, which, as you may imagine, is a major source of uncertainty, as it depends on both the Portuguese law and on the European emissions trading systems. Based on the free cash flows and required investment costs per project, the net present value, the internal rate of return, and the payback period were calculated and compared. Our preliminary results indicate that although the post-combustion option in green here presents a higher total CO2 avoided in all the cement plants, it should be excluded as it presents the highest payback period, the lowest IRR, and the negative NPV, meaning that the projects wouldn't be economically viable as the cost might not be fully recovered and the minimum rate of return isn't achieved. On the other hand, the positive NPV obtained in the reference scenario represented here in gray and with oxyfuel combustion in blue is a clear sign of economic viability, as it determines that on top of recovering the investment and fulfilling the minimum income required by investors, a financial surplus is generated. At this point, it seems that the oxyfuel method should be implemented in the pilot CCUS plant applied to the Portuguese cement industry. So in conclusion, the cement sector generates 7% of anthropogenic CO2 emissions. The CCUS is one of the most promising technologies to reduce them. However, it requires specific in-depth environmental and economical analysis to explore different pathways for its implementation in individual contexts. In particular, at C5 Lab, we are focused in defining criteria to assess the decarbonization potential and economics to retrofit Portuguese cement plants with carbon capture. For that, we've used a methodology divided into 5 main steps. As the cement plants are scattered through Portugal, by themselves, they don't define potential clusters. So we've identified 3 different areas in this region for the implementation of the CCUS technology: A, B, and C. Location A is the industrial axis between Lisbon and Setúbal, which includes Simporalhandra and Cecilotão. Location B is the northern part of the region with the Simporças plant. Location C is near Marinha Grande and includes the Cecil Cibrapataias and Cecil Maceira Leach plants. Afterwards, we've defined a reference scenario without CCUS, and we've identified 3 carbon capture methods: pre-combustion, post-combustion, and oxyfuel. But location C and pre-combustion were excluded as they presented the lowest CO2 avoidance potential for the cement industry. After that, we've performed an economic analysis to compare post-combustion and oxyfuel technologies for each cement plant. And the preliminary results show that oxyfuel combustion is the most economically viable considering the input parameters. However, we still need to conduct more studies to determine the best location, A or B, for the implementation of an oxyfuel carbon capture plant in the cement industry, considering CO2 storage, but also the potential integration with green hydrogen to transform the captured CO2 into high value-added products such as materials and fuels. So this research is far from over. Now, we will analyze different scenarios regarding the carbon taxes, the cement price, among with other variables. We will also include the CO2 transportation and storage costs, as well as the green hydrogen potential costs and benefits. Thank you, and feel free to ask me any questions now or contact me later through LinkedIn or by email.
Can you see my presentation?
Yes, please start.
Okay, good. So my topic is, what I'm working on currently is a polymeric material, which has property of being biodegradable in order to be used as implants in traumatology when you heal the broken bones. So as you might know, hopefully you don't, when you have a broken bone, you must, if it's some complicated damage, you have to put some fixating materials which immobilize, which means you make your bones not movable in order to grow them properly. Yeah. Otherwise, you just put cement like material on the broken part. And after the osteosynthesis is complete, which means when your bone is not broken anymore, you, in most cases, patients to be undergone second surgery, invasive surgery, which means you cut your patient again, blood, antibiotics, and so on, in order to remove that metallic implant. And this is disadvantage actually, because you're making a healthy person unhealthy again, because you're making a surgery. Yeah. And the solution is to make this implant biodegradable, so that it can dissolve, it can erode, it can disappear after some time. So here the green, it's just a photo how biodegradable implant looks like. So same shape, same function, but material is not metal anymore. It's a polymer, which is stiff enough, but able to degrade as after some time. I made analysis for my region. On regarding how many relatively simple surgeries in traumatology are made each year. So I had statistical data only about from 2015 and 2016, and the trend was obvious that number of surgeries are increasing just because of natural process. We have a growing economy, more buildings are built, more children are born, the population is growing. So naturally and unfortunately, more traumatological, more surgeries are necessary each year, are happening each year. I am not the smartest guy in this planet, so there are already solutions which I'm suggesting. Here at the right side, you can see the manufacturers of biodegradable materials for medical application. So you can see there are a lot, and some companies are very familiar to you. So it can be Toyota, Mitsubishi, Purak from UK, Cardiel, I think it's USA. Yeah. Purak is Netherlands, sorry. So those materials are FDA approved, so which means it's no carcinogenicity, so it doesn't cause cancer. So despite being synthetic, this material is not carcinogenic. It does not give some severe immunic reaction. It does not affect on fetus, and it doesn't have any chronic or acute toxicity. So it's not toxic. Chemical structure is like shown here. This is one of the materials. It's polylactic acid, the most popular. That biodegradable polymers are used widely in medical realm. So arthroplasia, ligament, tendon repair, prosthetic, reconstruct surgery, cardiovascular surgery, traumatology, orthopedics, urology. But I am interested in traumatology and orthopedics. So this is a photo, yeah, how it can be used. The broken bone can be fixed. What is advantages? I already mentioned. So the absence, the no need of reoperation, it's a main advantage. Actually, it's a main advantage. So you can save governmental money, you can save surgeons time, you can save patients insurance. So many advantages. So when you don't need second surgery in order to remove that implant. There is also a possibility, I will mention them later, possibility to make them individual. The implants itself, you can make them individual because you are using plastic, and there are at least 2 technologies which allow you to make custom-made shape plastic implant. Also, there is no stress shielding of bone tissue. Stress shielding means that it's a term which describes effect when the site of the broken bone becomes prone to damage because the tissue here, it does not accept stress during the implant presence and becomes more prone to damage because it's adaptive, our normal adaptive mechanism of the human body. Another example of stress shielding, it's astronauts after the trip to the space, where they don't in the space they don't have that stress on their muscles because there's no gravity apparently. So muscles became weak, so they adapt. There's no stress on muscles, so they don't have to be strong, so they became weak. And when they land on the earth again, they have to train their muscles again because there was no stress on their muscles. They should recover it. Same happens to the bone during the recovery when you have your implants. Similar effect. Also, you can chemically modify the molecular weight of the polymer. So doing so, you can vary the degradation time of your polymer. So the higher the molecular weight, the higher is the degradation time. So regarding the doctors, they don't have to undergo some special training because the shape of the implant is the same as a traditional metallic one. Same. So you don't have to make big changes in that procedure in a protocol, surgery protocol. Also, since it's not a metallic, it's not a magnetic, you can use computer tomography, you can use other like magnetic resonance imaging. It does not adhere, it does not cause any problems when you use this kind of imaging technologies. There is apparently additional advantages when we're talking about bone damage in children's and other extremum, all elderly patients. So if you think about fixation with implants in childhood, you for sure have to remove the implant because the bone is constantly growing. So you should not make any adhere this bone grow. You have to remove this implant. It means you have to make this second surgery. But if implants will disappear, you don't have to make this surgery. And not childhood, elderly person. The recovery processes are not as good as in more younger patients. So not having the invasive procedure, surgery procedure is beneficial. Also for future, you can immobilize some beneficial drugs into your polymeric material for antibacterial or for some other beneficial desired effects. The process of polymer degradation is well studied, and the polymer itself, it can be polylactic acid, it can be copolymer of lactic and glycolic acid, or it can be glycolic acid. Also, polycaprolactone also is used widely. These polymers, the monomers of these polymers are naturally present in our body. So the lactic acid, either glycolic acid, are naturally present in our body. So the only thing our chemical system have to do is just to make these excess substrates like lactic acid or glycolic acid to enter our natural metabolic pathways like Krebs cycle or Kelvin cycle and so on, and naturally can be eliminated from our body. From chemical point of view, the process causing this polymer to degrade is hydrolysis. It's a hydrolytic cleavage of the ester or ether bonds, and it is even more facilitated by different enzymes which are present in our body, like lysosomal cells, for example. So the good point that polymer itself is not toxic, and metabolites are not toxic as well. By varying molecular weight, by varying the technique of this implant obtaining, you can vary the mechanical strength of your final device, final object. So I haven't translated this, sorry, but it says that this is strength in megapascals. This is strength megapascal. So stress at which polymer or titanium product or bone breaks. So for bone, it's just 50 and 70 megapascals. It's for horizontal and vertical stress accordingly. For titanium, it has excessive stiffness. It's not bad, but it is just excessive. It's a 900 compared to 50 and 70. For steel, it's 1000. But you can obtain comparable stiffness for PGA and polylactic acid. PLLA, it means poly-L-lactic acid. It's a levorotary isomer of lactic acid. So it's also good news for us. So first, as I mentioned, we can vary the time. It's not shown here. We can vary the time of degradation, and also we can vary the strength of the implant itself by varying the polymer properties, which can be done at either at stage of polymer synthesis or at stage of this polymer processing, the mechanical process of obtaining the polymers. There are a lot examples. There are so many examples when biodegradable implants were used. I just took a random 3 because there are too much examples. I took these pictures from this article. So you can find the reference at the bottom. So 12 years old, 11 years old, and I don't know. I guess 9 years old patients. You can see the X-ray of the trauma. so broken bone here. And after 3 months, you don't see any problems. The bone is fixed. And for this process, for fixation, proper fixation of the bone, biodegradable polymer was used. Process of obtaining of the polymer which I've chosen was injection molding. This is GIF animation showing the principle. I have a mold, I call it matrix. I have a hopper, and here the heating process happens, and the molten plastic is injected and compressed into the mold matrix. This is other animation showing the principle. Yeah, this is not my animation. I've taken it from the YouTube, but it's very exemplifying the principle of injection molding. Let me skip some process. Yeah, see, the molten plastic is injected, and then it's cooled down inside the matrix. Then you open the matrix and can eject your object. Why I'm showing this? Because I've done this equipment by myself. I mean, I'm not the only. I have a team. So with my engineer friend, we made the casting matrix. We made the molding machine. We obtained the prototype. We have a patent. We have an article, and we have a partner from Trauma Surgeon Research Institute. So I hope to have a in vivo test soon. This is how the equipment looks like. I think it's very simple. I would say even primitive. This is a photos how it actually looks like. So there's pneumatic system here, which clamps the mold. This is also a second pneumatics here, which ejects the molten plastic, which is heated here, and we have a 2 heating zone here and here. It's controlled by this very simple interface. Here I control the pressure in the valves. Yeah, it's just explanation what stands for what. This is very first prototype. Yeah, pretty ugly, I think, but I know how to improve this. What I plan to do in order to move further, I plan to use, initially I was thinking about synthesis of the polymers, because my background is in polymer science, polymer chemistry, so I can synthesize polymers. But then I realized that if I can synthesize a polymer, it doesn't mean that I should. For sake of the experiments, in order to save time, I think it's better to use polymers which already available from the shelf. Yeah, so simply saying, it's better to buy the polymers which are manufactured. Then I just can prove their structure. If they lack some information, I can get some more information about the material from TGH, from DSC, from GPC, confirm chemical structure, determine crystallinity, confirm molecular weight, then use it for the production. After that, I want to try 2 regimes of mold casting. So hot die casting and cold die casting. Relatively cold, so it's just room temperature. Obtain different various samples, then study the properties in TGA, DSC, look at the crystallinity, and also mechanical. I haven't included here. Yeah, I should. Mechanical properties, of course, because it is critical for our purpose to have mechanical properties comparable to the titanium, the metallic implants. So my plastic implant should be stiff enough in order to compete with metallic one. So this is a critical point, a critical mechanical property. And then in order to save some time for experiments in the future, I wish to collaborate with the computing modeling team to get the mathematical model which describes the results. So depending on which factors, what properties I get, confirms this mathematical model with empirical data, and have a kind of virtual ability to make a virtual experiments in the future. There is some letters of interest from different medical institutions, patents, publications, blah, blah, blah. What I have to do, mechanical test, I accomplished this mechanical test. I haven't done a preclinical test, and I haven't done sterilization, and I think about the gamma sterilization. That's it. Rahmet, shukriya, teşekkürler, obrigado, mulțumesc, merci, dankeschön, dziękuję. So if any questions, I will be happy to answer your questions, of course.
Yes, please. Please.
Okay, my title of presentation is using bioinhibitors to enhance the corrosion resistance of carbon steel in acidic solution, presented by me, Mahbouba Azadi, faculty member of Materials and Metallurgical Engineering, Semnan University, Semnan, Iran. As an introduction, I can say corrosion attacks usually results in dangerous damages in various industries, such as the petroleum, oil, gas, electronics, and painting industry, as you can see in this image. Thus, to lower corrosion, there are several ways, like selecting the right metal type, protective coating, environmental control, metal plating, design modification, and corrosion inhibitor. Thus, utilizing inhibitors is one way to protect materials against corrosion. However, most organic inhibitors have a limitation based on environmental hazards. To lower the environmental limitation, there are several investigations to study the effect of various natural and biodegradable compounds on corrosion reaction of carbon steel in acidic solution. These compounds include nitrogen, phosphorus, oxygen, sulfur, or other heteroatoms or polar group in their structure to adsorb on the metal surfaces. For an example, such a compound like this structure in this slide contains OSN cyclic group, carbonic group, and these groups can adsorb at the steel surface and act as an effective inhibitor. Since extract of leaves, seeds, roots of plants, fruit and flowers, or either expired drugs or natural oils contain such desirable compounds, researchers were attracted at these materials to use them as green inhibitor. As shown in the right-hand side figure, the utilized green inhibitor can be tea leaves, honey, or other materials. And the other figure shows the distribution of the research works that performed in the last 2 decades on natural products. As new studies, we used extracts of insects, dead bodies, such as Astralis larvae, leech, and hyalomatic as bioinhibitor. These bioinhibitors can be good candidates to enhance the corrosion resistance of carbon steel in acidic solution, and our results show it. Therefore, details of corrosion mechanism, adsorption models, the optimum concentration of bioinhibitors, and thermodynamical measurements would be discussed in this present. At first, the chemical structure of these insect extracts would be presented. All of them contain compounds that could be absorbed on the steel surface. The extract main compound of hyalomatic and larvae is chitin as shown in figure 1A and 2 respectively. And the main compound of leach extract is hirudine as shown in figure 1B. They have large molecules, and it is notable that all these results were published in various papers recently. Fourier transform infrared spectroscopy FTIR results for all these bioinhibitors are presented in these slides. Figure 3 to figure 5 all show that all bioinhibitors contain CN, CC, NH, OH, CO, double or single bonds. These results confirm that all utilized bioinhibitors can be absorbed at the steel surface and protected. To investigate the corrosion properties of carbon steel in acidic solution in the presence and absence of bioinhibitors, electrochemical impedance spectroscopy, EIS, TOFL polarization, and weight loss tests were performed. In this slide, TOFL polarization tests in presence of various bioinhibitors are presented in figure 6 to 8. For hyalomatic and leach extract, by increasing the bioinhibitor concentration from 1 to 3 grams per liter, the corrosion rates decrease. And for the larva extract, the lowest corrosion rate was achieved in the optimum concentration with a value of 1 gram per liter. It means when the concentration of larva extract increased more than 1 gram per liter, the inhibition efficiency would be decreased. This slide also shows the details of measurements from TOFL plots. The inhibition percentages, theta, shows that for the hyalomatic leaches and larva extracts, all of these bioinhibitors was about 62 to 95%. Besides, all of them act as mixed inhibitors since changes in the corrosion potential were more than 85 millivolts. It is notable that the test temperature was the room temperature, about 25 centigrade degree, and the acidic solution was 1 molar HCl and 1 molar H2SO4 solution. And the highest inhibition percentage was related to the hyalomatic extract as inhibitors. The EIS measurements were also investigated. Similar to TOFL plots, the corrosion resistance of carbon steel specimens in acidic solution increased significantly in the presence of all biomaterials. And by increasing the bioinhibitor concentration, the impedance increased too. It is notable that the EIS plots contain Nyquist plot, and the bode and phase plot. And shown in this slide, the highest impedance was belong to the hyaloma thick extracts with a concentration of 3 grams per liter. And this slide demonstrates details of EIS test results through utilizing a ZView software. The inhibition percentages, theta, for all utilized bioinhibitor was about 50 to 92%. For hyalomatic and leach extract, the highest theta was achieved in presence of bioinhibitor in concentration of 3 grams per liter. However, for larva extract, the highest value of theta was related to specimens with the inhibitor content of 1 gram per liter. Besides, the highest value of theta was related to the hyaluromatic extract. The schematic suggested electrical circuits are shown in this slide. When the bioinhibitor adsorbs at the still substrate, changes in the double layer properties would be occurred. And when the bioinhibitor concentration was adequate, it could be formed and separated layer at the still surface, as shown in figure C. In this situation, two circuits would be appeared in the suggested electrical circuit. Otherwise, one circuit would be suggested for electrical circuit in presence and absence of bioinhibitor, as shown in figure A and B. The effect of a long time immersion versus the corrosion rate of carbon steel in acidic solution is also investigated in this slide. All plots showed that the weight loss increased by increasing the immersion time. However, by presence of bioinhibitor in the corrosive solution, the corrosion rates decreased significantly compared to the blank specimens without inhibitor. All plots also showed a linear behavior for almost specimens. The thermodynamical calculation were also done as shown in this slide. For all bioinhibitors, free energy was negative, and this value and the value of it was about 20 kilojoule per mole. The value, or this value, showed that a physical adsorption of molecules at the steel surface would be occur. For larva extract, this value is the minimum among other extract. Besides, all of them showed long Muir model based on the related plots. It is noticeable that the negative value of free energy showed that the adsorption of extract molecules at the steel surface was spontaneous. Field emission scanning electron microscopy, FESEM images of the steel surface in the presence and absence of all these utilized inhibitors were examined. The defects such as a pore, pits, cracks, or other imperfection decreased in the presence of bioinhibitors significantly. Such events showed lower corrosion attacks at the surface of steels when bioinhibitors were adsorbed at the steel surface. The concentration of bioinhibitors in this slide was optimum for each solution, and the immersion time was about 12 to 72 hours. Atomic force microscopy, AFM images of the steel surface after 1 hour immersion in the acidic solution in absence and optimum concentration of bioinhibitors are presented in this slide. The roughness of corroded surface decreased significantly by presence of bioinhibitors. This reduction was about more than 50%. The lower roughness was a result of lower corrosion attacks of corrosive hydrogen ions at the surface. And as a conclusion, I can say that both EIS and TOEFL polarization data at the room temperature indicated that by adding bioinhibitors, the inhibition efficiency increased significantly. The optimum concentration of all these utilized bioinhibitor extracts was about 1 or 3 grams per liter. Based on the electrochemical test, the adsorption isotherm was the Muir model, and in this model, each site of steels for corrosion reaction was occupied by 1 atom of inhibitor. All bioinhibitors extract molecules acted as a mixed inhibitor due to the corrosion potential. FESEM images showed that the degradation of the corroded steel surface after 72 hours in acidic solution with optimum concentration of extracts reduced significantly. The roughness of corroded surface after 1 hour decreased based on the AFM images, since extract as the physical barrier for corrosion ion diffusion. Based on experimental result, a proposed inhibition mechanism consisted of the physical adsorption of inhibitor molecules from the N or O side at the steel surface and create a barrier layer for hydrogen ions. And the most important utilized reference for preparing this presentation are reported in this slide. And I thank my team for their work. And at last, thank you for your attention. And if there is any question, I will answer it.
Dear colleagues, thank you very much for your kind invitation to present my presentation and few words. My name is Anatoly Zavdoveev. I am a senior scientist at Photon Electric Welding Institute, and today I will make a report with the title SPD as a powerful tool for nanostructure fabrication in low carbon steels. SPD, this is abbreviation of severe plastic deformation. What does it mean? Severe plastic deformation is the kind of deformation which gives us opportunity to accumulate plastic strain in the material, and this can lead to significant grain refinement. And as you know, according to whole patch law, if we got grain refinement, we achieve improvement in strength characteristics. So let's consider more detail the effect of severe plastic deformation on low carbon steels. One of the most popular severe plastic deformation method is so-called twist extrusion. What does it mean? It means when we take prismatic billets and it pushed or pressed through a twist channel. Here in twist channel, the plastic deformation is accumulated, and the dimension of billets doesn't change. This gives us opportunity to accumulate from pass to pass a very high level of deformation or strain. And this leads to grain refinement as well as improvement of properties of material. And the most important advantages of SPD method between conventional metal forming is that if we use, for example, conventional metal forming, such as drawing, stamping, the dimension of specimen is changed, and it can be elongated or it can be simply change significantly the dimensions. In this case, you can't accumulate strain infinitely, but when you use severe plastic deformation, the dimensions of billets doesn't change, and this is a great advantages of such methods. So here we can see the microstructure of low carbon steel after twist extrusion, after severe plastic deformation through twist extrusion. Left side, you can see optical microscopy photographs, and right side, you can see scanning electron microscopy in same mode, where you can clearly see pearlite colonies in different magnification. So if we look optical microscopy, we can see that pearlite colonies are grain refined significantly and uniformly distribute through the perpendicular or through the sample. And pearlite colonies are also significantly grain refined. And additional advantages is we have changes in morphology of pearlite colonies. If here we have lamella-like type after twist extrusion or severe plastic deformation, we had globular one. The next one very interesting effect is that after twist extrusion, right hand, we can see that the volume number of high angular grain boundaries significantly improved, and the values of last one is increased. And high angular grain boundaries will determine the strength characteristics of material, in our case, low carbon steel. And here with green color, we can see distribution of so-called Schmidt factor. It shows us the distribution of deformation, or other words, the number, or not number, the strength of Schmidt factor will show us in which grain the further deformation will start more easily. And next one, we can see the effect of twist extrusion on recrystallization. Here we can see three types of recrystalline grains. First one in the bulk of the grain. Second one is absorbing neighboring grains. And third one is on grain boundaries. This is a three type of recrystallization. In our case, it will be dynamical recrystallization due to these grains has appeared during plastic deformation. And all figures of recrystallized grains show us the following effects. Left side of figure, we have initial structure or initial texture of recrystallized grain. We have 1, 3, 4 peaks of recrystallized grain. All figure for twist extruded sample shows us that these poles are not appear here. And here we have exactly different pole figures peak. It tells us that these grains, recrystallized grains, are uniform grains. So this is the evidence of dynamical recrystallization during severe plastic deformation in low carbon steel. Here we can see the effect of twist extrusion or severe plastic deformation on mechanical properties. And very important and should be emphasized that physical properties such as density is almost doesn't change. This is a very important and very interesting because if you use conventional plastic deformation method, for example, elongation or pressing or something, the density of deformed metal will be increased because the accumulation of defects are in progress during conventional treatment. But using severe plastic deformation, we have the constantly almost doesn't change in density. This is due to specific characteristic or specific peculiarities of severe plastic deformation, where the Archaea defects are healing, and also due to significant grain refinement, we have significant improvement in strength from 800 MPa to 1020, 200 MPa. Also, the micro hardness is almost 1.5 times greater. So let's consider another one severe plastic deformation method is a continuous method, and it calls rolling with shear. This method incorporates the knowledge of shear deformation and the knowledge of equal channel multi-angular pressing, where in deformation zone during rolling with shear, it is realized scheme of deformation of SPD deformation. So such deformation scheme give us opportunity to form uniform equiaxial grains after rolling. This is very important because when we use standard rolling, we have elongated grains, but when we have shear deformation in deformation zone, our grains become equiaxial, and this will be inherited during further deformation. Also, when we use rolling with shear, we have much more higher so-called technological plasticity or a reduction to failure. It equals 70% instead of 48% for standard rolling. And here we have the structure, the microstructure of samples of low carbon steel after rolling with shear. And you can easily see the difference between left and right. Left hand we have rolling with shear effect, and right hand is the classical traditional rolling. So you can see that left side we have uniform equiaxed uniformly distributed grains, and right hand we have as small grains such and big grains with different orientation, and the effect of this severe plastic deformation method on Schmidt factor also gives us clear evidence that during rolling with shear, the deformation distributed in the samples more homogeneous, and when further deformation will apply, the whole sample will deform uniformly instead of standard rolling sample, where more green grains will start easily and more white grains will be not deformed. And here we can see also that the number of high angular grain boundaries in rolling with shear much higher between the standard rolling samples. This also lead to more great or not great, more improved strength characteristics. Also, we can see that the volume number of recrystallized grains are also higher in the case of rolling with shear. When we consider the thin structure using transmission electron microscopy, we can see that if we use standard deformation or standard rolling in the left side picture, we have grain boundary and the body of the grain with the high density of dislocations. And when we use rolling with shear, we have also grain boundary, triple grain boundary, but it is a little bit wider than in the standard rolling case. Also, we have the body of the grain almost clear from dislocation. Why? Due to this dislocation, you can see the traces is flow to the grain boundary. This is additional resource of plasticity for such material, for such deformed material. And owing this effect, we have much higher plasticity for this low carbon steel after rolling with shear. And it will give us opportunity to further deform this material for much higher accumulated strain. For example, here we can see the changes of physical properties such as density and strength properties such as micro hardness, ultimate tensile stress, and plasticity in terms of number of bands before failure. You can see that black solid lines reflects effect of rolling with shear. And here we have that accumulated strain, further strain of conventional drawing for rolling with shear gives us possibility to accumulate almost 1.5 equivalent strain, while for standard rolling, we have a little bit higher than 0.5. And this is owing to specific structure formed during severe plastic deformation. Also, the density, physical density, as I told for twist extrusion, for rolling with shear are also remains almost constant. And these results are closely connected with the results of optimal tensile strengths. We have much higher tensile strength for rolling with shear material and much higher the total number of bands for rolling with shear material. And reached strain and equivalent diameter of the wire is equal for standard rolling technology and conventional drawing is 1.8 millimeters, while in the same, almost in the same condition, but with more higher accumulated strain for rolling with shear technology, we can achieve 0.8 millimeters diameter. This is amazing results. And also we can see very interesting effect. This effect shows us that while we drawing sample, classical sample, we have the following regularities. The grain of the perlites, ferrite and perlites are continuously increased. But when we use rolling with shear technology, the grain boundaries are increase, decrease, increase, decrease. And this effect will give us opportunity to reach much higher level of accumulated strain. And also here we can see texture and the peculiarities of texture. For rolling with shear, we have significantly expressed blurring of the texture. For example, upper figures reflect classical texture of drawing when all grains are in the same direction. And when we use rolling the shear, we have the intensity of poles almost 2 times lower, and they are bluer. It means that the grains are rotate from the drawing axis on some angle. And also very interesting results showing the distribution and evolution of nanovoids, nanopores is a small angle neutron scattering, which shows us then using rolling the shear technology, we have the size of particles or nanovoids becomes to saturation, while the using of standard technology, this size continuously increase. And these results correlate with results for conductivity, electrical conductivity for these materials. And we also know that during classical deformation, the conductivity continuously increase or resistance is increase. But when we use rolling the shear technology, we have saturation and the plateau of conductivity. We can accumulate strain and conductivity will remain. This is also very interesting result. So severe plastic deformation is a powerful tool to achieve and improve physical and mechanical properties of low carbon steels. Thank you very much for attention. Please, your questions.
Yeah, a very good afternoon. I do not know all the audiences may be from different parts of the world, so it's better to address the audience with without mentioning any time. So today's topic is my novel CVD growth method of diamond materials for advanced science and engineering applications. I'm Avdesh Malik. I have been working with diamond materials for the past many years. And today I'll be presenting some of my recent experimental results. Before I start my talk, I would like to invite the audience about the, I'm editing a special issue of NDPI journal Energies. It is Materials for Renewable and Sustainable Energy. If you scan this, then you will be able to go to the page and get more details about the special issue. And the deadline of submission is January 2022. So, and it has a very good impact factor of 3.004. So there's a good opportunity for the audience to publish in high impact factor journal. So my talk can be divided into these segments like introduction, motivation of my research, the materials and methods that I have used, the results that have obtained recently, and I will try to discuss the different aspect of the obtained results with a conclusion at the end. So as you know, that diamond is present under the ground and so from mines, people are procuring diamond which are formed billions of years ago. But there is a recent development in the manufacturing technology in the chemical vapor deposition. That process in 1980s allowed scientists to grow diamond in the laboratory. There are other methods also for the growth of diamond other than the naturally available diamond, that is high pressure, high temperature. Today's focus will be CVD growth of diamond. So if we go, if we try to compare different aspect, like historical aspect of this lab-grown diamonds, we are calling it now laboratory grown, and the diamond which are natural from mines. So the mine diamond was in the 9th century BC. Only India supplied diamonds to the world from alluvial riverbed deposits and mines. And from 15th century, there was a in Antwerp, Belgium, yeah, there was polishing activity started. And later on, in 20th centuries, the diamond business was dominated by De Beers. So this is the history of the, means a very uh, milestone wise, like it is not, it's a very long history. But the technological laboratory grown diamond is not such a long one. It started when GE, General Electric, discovered diamond synthesis by high pressure, high temperature in 1954. And then that was a very great effort, like mimicking the high pressure and high temperature that exists under the surface of the earth in the mantle and the crust. So they replicated with manufacturing of some anvil instrument, and that enabled to synthesize diamond. But that process, that experiment, means those instruments are difficult to fabricate, and the quantity, the volume of diamond that is synthesized is not also very high in amount. But in 1980s, in Japan, there is a National Institute of Material Research. They developed a process by CVD, chemical vapor deposition, which helps to grow diamond very fast. And that technology has evolved over the past 40 years, and which has now made possible for the industry to adopt CVD technology for making jewelry. Diamond jewelry, means the diamonds that are made by CVD are, in fact, the De Beers, who dominated the natural diamond business in 20th and 21st century, they have started this CVD grown diamond company also, that is known as light box jewelry. So it is a now established process for the industry also to grow diamond. Now, there are many positive and negative aspect of mining, like mining is not environment friendly. Mining damages, yeah, there are child labor issue, there are smuggling issue in poorer countries. Other than these ethical issues, it is also environmentally not sustainable. It damages the environment. Whereas lab-grown diamond is unlimited source. It is sustainable. It is environment friendly, low cost, all the ethical practices. So there are manifold advantages of lab-grown diamond. Now, why diamond is an important material? Diamond is the hardest material known to scientists. If you compare different hardness values in GPA, you will find that the diamond is much harder than the material, the industrial material like steel, alumina, titanium carbide, which are widely used material in the industry. Compared to that, diamond is very hard. But the availability, like the steel is available in huge large quantities. But diamond is not available in such a large quantity which humans can use for their advancement, for their different applications, because diamond is costly. So that is why it is used as jewelry. But nobody can think that diamond, the same diamond which are used in jewelry, to be used in the technology, like in your smartphone or in your laptop, because diamond has a very high thermal conductivity. The high thermal conductivity makes the heat that is generated within the electronic devices or the laptop computers to dissipate away easily. So that makes the electronics to operate in a much faster way, which we'll discuss later in the later slides. So another aspect of diamond is it has a very strong covalent bond, carbon-carbon bond among all the allotropes, that also make it a very stiff material with high Young's modulus. If we compare the Young's modulus of different material, we'll see that diamond has a 1220 GPa value, whereas the steel has 200. So it is much superior to other materials. It is also, as I was telling about the thermal conductivity value, it is a very high thermal conductivity material, the best among anyone. Copper, if you compare that with copper, if in the scale of cooling ability, if water is 1, then diamond is 2000. Obviously, the unit is watt per meter Kelvin. And with this superior cooling capacity, it is used in the electronic chips like thermal chip. If this is the diamond heat spreader and there is your electronics, it is used in the integrated circuits for the heat dissipation of the, because current is flowing, electrons are flowing, that is making resistively heating this circuit. So that heat that is generated has to be thrown out, and diamond is the best material for that to do. Yes, now so as we know, the silicon is the semiconductor material. It has a well-established market, but the limit, the Moore's law, is telling that the density of transistor will double in every 2 years. To keep the pace with the Moore's law, silicon is not able to keep that pace. So there is requirement of the alternative material, or there are requirement of alternative design, which will help to maintain the Moore's law. So as you can see, the sizes are decreasing. This is the typical size of 200-volt silicon device, whereas 200 volts identical capacity gallium nitride device is such a small. So in such a small area, if the same amount of current is flowing through, so it will effectively heat the devices enormously. But that heat has to be thrown out of the device, otherwise it will not operate efficiently. Yes, other than gallium nitride, there are also other materials like silicon carbide, diamond, which are having very high band gap. They are known as wide band gap materials. As if we compare these wide band gap materials in this table, we'll see their band gaps are quite high compared to silicon. They have a very similar kind of property, but if we look at the thermal conductivity, diamond has a very high conductivity. Even that if the diamond is not so pure like single crystal, then also it has a very high thermal conductivity compared to other wide bandgap material. Gallium nitride has a Wurtz's crystal structure, and also it has zinc blende crystal structure, and diamond, which also had the same zinc blende structure. It is nothing but two interpenetrating FCC lattices. So, but if we try to integrate diamond with gallium nitride power electronics, there are some challenges. Like diamond and gallium nitride are two materials with considerable difference in thermal expansion coefficient. 1.2 is the value of alpha for diamond, whereas gallium nitride has a value of 3.2 per degree Celsius rise in temperature. Whereas in the elastic modulus of diamond has a value 10^12, which is very high, but gallium nitride does not have such a high Young's modulus. And the Poisson ratio is also quite different. And this leads to a material compatibility issue when we try to grow diamond by CVD over gallium nitride. So the motivation for growing diamond over gallium nitride is one of the wide bandgap material which is trying to replace silicon in the electronics industry, but the size, it has the problem of heat dissipation. So if we integrate diamond with the gallium nitride, then diamond having high thermal conductivity can dissipate the heat very effectively for the power electronics, gallium nitride based power electronics. So this is the motivation of my present work for the better thermal management of diamond with gallium nitride. So now the question is, with the motivation of my work known, then how should we approach the integration, means the combining diamond with gallium nitride? One obvious approach is to bond them together, like gallium nitride and algan can be directly bond with each other, like with freestanding diamond substrate. There is a work by DC Dumka et al. they have published a work in 2017 where they try to put the single crystal freestanding diamond with gallium nitride. The other approach is to grow gallium nitride based HEMT structures directly on gallium nitride diamond substrates. But the deposition of another means the approach that we are taking is growing diamond on gallium nitride. But it has a problem, means while we try to grow diamond by CVD, it has a very high temperature and low pressure. Such a high temperature, low pressure environment with microwave plasma. We also use microwave plasma. So plasma environment, the CVD growth environment, chemical vapor deposition with plasma is very detrimental, is very harsh for the gallium nitride. Under such a chemical vapor deposition environment for diamond growth, it makes the gallium nitride susceptible to etching. So under CVD diamond conditions, this is the typical reaction that takes place at 800 degrees Celsius inside the chemical vapor deposition chamber. So this work has been published by Professor May in Diamond and Related Materials Journal. So what then the scientists do? What do they do? They try to separate the gallium nitride from diamond with an intermediate layer. These are the cross-sectional microscopy, which shows that the usual practice in the industry or by the scientists, by the laboratory experimentalists, is to separate the gallium nitride for diamond with this nitride layer. It can be silicon nitride, it can be aluminum nitride, but the idea is to separate them together with a buffer layer. If we do not have any buffer layer in between in this picture, there is a erosion or etching of the gallium nitride due to the plasma of the diamond growth environment. There are many attempts by many researchers in since 2001 to grow diamond on gallium nitride. The recent one is to grow by argon chemistry. It was possible to reduce the temperature up to 400 degrees Celsius. So if we reduce the temperature from 800, that is the normal processing temperature of diamond by CVD, to 400, it is possible to avoid the etching of gallium nitride. So we try to do that. That is to reduce the diamond growth temperature. Usually the resonant cavity CVD, this is the resonant cavity CVD, it has a plasma which is spherical and confined within a definite volume. It has a very high processing temperature, 800 degrees. We have already seen that that is not conducive for the gallium nitride. It etches the gallium nitride substrate. So what we have used in our experiment, we have used a linear antenna CVD, which allows us to grow diamond at a very low temperature. So we have used the special kind of reactor for the diamond growth. So in our earlier work within our group with that resonant cavity, we have seen that these are the different layers, intermediate layers between silicon, then gallium nitride, then algan, then PCD. It is a multi-layer heterostructure. But our idea is to combine gallium nitride with PCD directly without intermediate layer. Intermediate layer itself is a hindrance for the heat dissipation. If we try to dissipate the heat effectively, then there should not be any barrier layer, buffer layer, that which people have been using. The very idea of putting a buffer layer in between gallium nitride and diamond itself is against the idea of effective heat dissipation. So we tried to reduce the diamond growth temperature and directly bond with gallium nitride. So how we did that? We used a innovative plasma reactor, that is a linear antenna CVD reactor, which made us to reduce the growth temperature. So removal of the motivation using this LACVD, we call linear antenna CVD, removal of intermediate buffer layer which scatters the heat, and also low deposition temperature will reduce or even exclude the plasma impact on GaN. These are my unpublished results, so please do not share anywhere else. So before, this is a sample GaN on sapphire, then we Yeah, this is the surface plasma pretreatment before CVD growth. The surface plasma changes the pH of the, means it helps the subsequent nanodiamond seeding. This nanodiamond seeding, it is a suspension in water, so it has certain pH. So for the effective seeding of these samples, it has to be seeded with diamond nanoparticles. To enhance the seeding, we have made a surface plasma pre-treatment. So after seeding, we dried the water and then we put the samples inside the CVD chamber. You can see these are the samples, and these are the typical processing parameter, like the temperature window is no more 800, it is less than 400. And then microwave power in between 1.5 to 3 kilowatt. Gases, we use hydrogen, methane, CO2. Pressure, we try to keep very low of 0.2 millitorr, and that is how we grow nanocrystalline diamond. This work has been published in this journal paper, APL. So after growing diamond, we saw that this is the typical Raman spectra with 89% hydrogen, 5% methane, 6% CO2. The TPA, the trans polyacetylene peak, is at 1484, and the sp3, that is the diamond sp3 peak, is at 1332. And these are the top diamond layer, and there is a bottom gallium nitride substrate. So we could see that bottom gallium nitride substrate was also producing well-defined Raman peaks. That implies that the underneath gallium nitride substrate was not damaged by this low temperature CVD growth. We have additional proof also from FTIR, XRD, different confirmative results, which tells us that the gallium nitride is not etching with our optimized process recipe. And to optimize the parameters, we varied the distance between the quartz tube and the substrate from 3 to 15 centimeter. We changed the microwave input power also from 1.5 kilowatt to 2.8 kilowatt. Also, we means the microwave input power mode was either continuous or in pulse mode. For pulse mode, we typically use 20 kilohertz, 45% duty cycle. So thereby we could see that the optimum processing condition was achieved with a well cauliflower-like NCD morphology, nice FTIR peak, nice Raman peak for diamond. It has a high FWHM value, but still it was nanocrystalline, so it was very much expected. But if you compare the height of the peak of sp3 with the non-diamond, that is graphitic deposition, it was a very high quality diamond that was deposited at the optimum processing condition. Therefore, we conclude a combination of intermediate microwave input average power of 2 kilowatt and a pulse mode frequency 20 kilohertz, 45% duty cycle was used to achieve low substrate temperature while maintaining high active atomic hydrogen species in the LACVD plasma environment. This was achieved by in situ lowering the substrate stage to a distance of 6.4 centimeters away from the linear antenna after starting the CVD run with a distance from 5 centimeters. This was something innovative we try to achieve. And such optimum variation of antenna to stage distance and reduce power level could produce high intensity diamond Raman signals for NCD on gallium nitride films without substrate etching. I must acknowledge this work was done at Hasselt University in the group of Professor Heinen. He's the group leader, and this is the dinner lunch we had. Yeah, it was quite late, 5 o'clock, 6 o'clock. So we decided to go for lunch, but it was yeah, quite late. Anyway, so yeah. I have been living in Hasselt, nice place. Thank you for your attention, and yeah, don't forget to submit a paper in this journal, special issue of energies, materials for renewable and sustainable energy. We can also give concessions Yes, thank you. That is all I had to say. Now you can ask me questions if you want.
Fazal Ilahi. So let me share my screen right away. So let me introduce myself. I'm a postdoctoral research associate at the University of Delaware in the Material Science of Engineering department. But my research is, most of the things I'm going to talk about today is the mechanical properties of material. That's what my background is. My BS and my PhD was in mechanical engineering. So what we do is we try to investigate different ceramic composites for different applications. And our purpose is to increase the toughness of the material. So I'm going to talk about more. And so today's talk is mostly analytical and simulation based. I'm going to introduce maybe a few experimental evaluations at the later stage if I have some time. So let me go straight away with our the motivation and introduction, like why we're doing this. So in the video, you can see, let me start my laser. So you can see here we have the space shuttles that the NASA used in like the before 2010. So they used to carry ISS supporting staffs and different servicing tools. So what happens is in 2003, there was a big disaster with Space Shuttle Columbia that happened in Texas. So when a space shuttle was returning to the Earth, it hit a very small earth particle, and that the entire space shuttle was disintegrated. And so when NASA investigated that, they found out quite a few things. So their finding was that the material was not strong enough to hold the heat that the space shuttle was experiencing when it was returning to the Earth. So the three reasons they came out: that there was some crack and the temperature was really high, and the protection system that the space shuttle was using was like carbon-based thermal protection system. So that's why they provided like millions of dollars to a lot of the universities. So we are one of the team that are working on developing materials for extreme dynamic environments. Those materials can be used in space shuttles or maybe nuclear reactors, turbines, or even automotives. So our task is mostly to improve the fracture toughness of such materials, which held the crack propagation or maybe carry out the service life of the material for longer time. And then we are trying to develop some novel materials as well, which are not being used so far. And we also want to develop a simulation framework at the atomic scale. So I think all of you know that today's world is nanoscale world. So everyone wants to know what is happening at the nanoscale. So that's what we want to develop, and we want to give it to our funding agency. So our starting material is silicon carbide. So I'm just saying why we started with working with silicon carbide. I think Dr. Malik was trying to explain a different perspective of silicon carbide, which is a really important material right now for semiconductors, different electronic devices, for transducers, and even for like mechanical applications. So the reason that we are interested in silicon carbide is, you can see here the toughness of silicon carbide is low, but you can see the strength is really high. It's almost as high as like more higher than glasses and even a lot of more metal alloys. So because of that higher strength, we want to work on it. And the annual growth of silicon carbide, you can see it's really getting higher and higher every day. So our investigation is just based on silicon carbide. We are not working with any other materials. And also just for a fun, is the new iPhone 13 is silicon carbide coated on the top, just to have more break protection. Yeah. Okay, so what we do on silicon carbide, so we study two different parameters. One is if we have, say, a simple substrate, and there is no crack or no discontinuity in the material. So given that the material is pristine. And the second condition is if we have a defect in the material, maybe a crack or maybe the vacancy or any kind of defects. So in these two types of systems, if we load them along, say, Y, so just think about uniformly tensile testing or uniform loading or straining. So what will happen to the material? If there is no crack, the material will break, and if there's a crack, the material will break. But how much? Our intention is to stop the crack growth, or maybe stop the crack nucleation. So here I'm showing you two. Okay. So these two are two illustrations. So these two are the same material with crack. There is no other defects available in the material. So you can see in the animation that both of them have cracks, but both crack propagations are not same. So both those two are the same material, same simulation environments. Crack length is same, everything is same, material is same. So but we found out for different materials we could find different crack propagation patterns. So for the first one, crack is propagating straight. The second one, crack is kind of branching out and making a V like this. So our intention is to investigate that so that we can suggest how we could stop the crack growth or maybe delay the crack growth so that the service life of the material increase at the end of their life. Okay. So to do that, what we do is we use molecular dynamics is the first thing that we used to investigate the structure. So we started with 3C silicon carbide, which is the diamond cubic structure. Now, so if we investigate the crack pattern that we wanted to see, so let's see if we have a crack here in our the second case of the crack and non-crack system. So if we have a crack, how the crack tip would look like in terms of atomic structure. So if we investigate that, we see that for if the material is grown along 1-1-1 plane, the crack tip looks like this. So all the atomic bonds, they are kind of parallel to the loading directions. And if the material is grown along 1-0-0 direction, which will be, so you can see for 1-0-0 structure, this material is that the crack tip is kind of a V like this. So the atomic bonds, these are all the atomic bonds. So those bonds are creating an angle, and for the 1-1-1, it's kind of parallel to the loading direction. So that's why those two crack propagations are different. Just for the crystal structure, there is no other factors required for the crack pattern to be different. So that is kind of a very low scale investigation. These are really important when we are seeing a crack propagation in macro scale. We try to investigate, or we want to visualize that what is happening at that scale. So this is what our reasoning that why silicon carbide shows different crack patterns. And now my question is, so just for your thinking, now let's think of our phones. So if a phone falls from our hand, you can see all the crack that are nucleated and propagates, they are very abrupt. They are kind of, you cannot find a pattern. So my question to you is, why do you think that the crack pattern is that much abrupt? So we can talk about it. Just think about it. We'll talk about it later, and you can ask me questions. So let me go to silicon carbide again. So what we do is we're trying to come up with composite models with different compositions of matrix and inclusions. So what are the matrix and inclusions? So if we take, because we are doing atomistic simulations in molecular dynamics, they are really computationally expensive. So for one simulation that I showed you before, just for a fun fact, so that one simulation takes around, say, maybe 3 to 4 days. So it's really computationally expensive. So what we want to do is, so we don't simulate the entire composite with many fibers in it, what we do is we take one representative volume with one inclusion, or you can say one fiber and just array of matrix. That's it. So this is our atomistic model. And for that inclusions, we are trying to incorporate different amount of nanowires, silicon carbide nanowires, which are commercially available. So we buy them, we can actually grow our composite in our lab, and then we can simulate it in our computer. So that's what we are trying to do right now. So for that purpose, just to have experimentally coherent simulations, we are using nanowires as our fibers. And the matrix material will be silicon carbide as well. So the cubic silicon carbide is the matrix, and cubic silicon carbide nanowires are our inclusions. So this is just a model, like the 2D model, just to understand better what we have. We have a nanoparticle inclusion, and then we have the matrix, which is silicon carbide. We have a crack, and we want to load it along the Y direction. And then we want to see how the crack propagates, and when the crack interact with the inclusion, what happens. So because I showed you before that crack can propagate two ways. One is it can go like bifurcating, and also it can go just propagate straight. So we want to see that what happens when the crack hits that inclusion, whether we can delay the crack propagation using that inclusion. So that is our purpose. Okay, I'll discuss the numbers later if you want to know. Okay, so let's study. Let me show you the debonding and fracture, because we have the interface. So I think a lot of you talked about the interface just the previous talk. So let me talk about the interface a little bit. So this interface is really kind of theoretical for now. Because when we model it in the atomic model, so both of them are silicon carbide, both of them, their lattice structure is diamond cubic. So we didn't have to worry about the interface that much, but still, I mean, I think I spent at least 6 months just to cure that interface, even in the simulation. So it's really difficult. And also just to remind you, all the simulations that I'm showing, they're for 1500 Kelvin. So which is like really high temperature, and the chances of debonding, chances of failure are really high at that high temperature. Okay, so let me show you one. Okay, I don't know. Can you see the video? Yeah, so this is what happens if we load it from the Y direction. So the debonding happens through the interface, which is really intuitive, because I mean, we all know that interfaces are really the weaker points. Even if we kind of cure the interface and increase this strength a little bit by different ways, but we could not get that debonding array. So when you load it, always material failed from here. And if we see this is very interesting. If we see that what happens to the atoms at that interface. So this atom is in the like the orangish color. This is the weakest atomic bond in the interface at that moment. So what happens is when we apply loading, that bond break first, and then it just the crack is just propagated along two ways. It's really quicker. I think I can show that. Yeah. So can you see the video? Yeah, I think you can see that.
Yes.
Yeah, so it's really very cool to see how that one breaking bond actually tear apart the entire structure. So silicon carbide is a brittle material, so that's why crack propagation is really faster. It's just opposite to the ductile materials like polymers or gold or maybe any other metals. So it's really quicker. So that's the problem with silicon carbide. That's why that's how the space shuttle that I showed, when only one crack built up on the wing, it fails within maybe 30 seconds, because the brittle material cannot hold the crack for longer time. It's going just very quickly. So this is just the toughness changes, how the strength change with the volume inclusion. So this is also intuitive. So if we increase the surface of the inclusion, of course, the toughness will go down, because the amount of that overall toughness of the material, or the debonding toughness of the material. So let me show you what happens to the crack case. So this is one crack case where we have a crack, we have a nanoair, like a nanoparticle, now with a certain diameter, and we have a matrix, and we are loading it. So what happens to that is, so you can see the video, the crack comes in, sorry. So crack comes in and it interacts with the nanoparticle, and then it crosses that interface again. So there are two interfaces that the crack has to pass. One is when it reaches, and one is the passing point. So, and in between it has a nanoparticle to break, which is silicon carbide 100. That means the crack will bifurcate because of the bond angles. So that is what happens. And for a smaller diameter, so these are the two different diameter cases that I'm showing. We considered maybe 20 of them. So for the smaller diameter, you can see that the crack is stopped for some time. I don't know if you, so I'm showing you again. So for the smaller nanoparticle inclusion, the crack comes in, crack stops here for a moment, and then it starts propagating again. So in these two cases, the first case, the size of the nanoparticle is larger, but the crack never stops here. It comes in, it crosses the nanoparticle very quickly, but for smaller one, crack stops. So what does it mean? It means that in our investigation, we found that if we can use like small enough nanoparticles, that will be much more helpful, which is a little counterintuitive from the literature that we had. So we are still fighting to publish it. It's under review for maybe 6 months now, in the 3rd revision. So that work is really it changed a lot of our intuitions, and it helped us to understand how we can develop the toughness of the material. So in the numerical values of the toughness, when we increase the volume inclusion, because the nanoparticles are much more stronger, and it can increase the toughness. So the size of the particle increase the toughness of the material or the strength of the material, but it cannot hold the crack. So the crack propagation is delayed with smaller particles. So these two are like the opposing properties of a material. So we are trying to optimize that, and we found a few optimized conditions, but because of that NDA that we signed, I'm not able to show that. But yeah, but I can answer if you have any questions. So let me show you the outcomes first. So the two outcomes that we have is, we have the debonding toughness. If we increase the size of the nanoparticle, the debonding toughness goes down. And if we increase the size of the nanoparticle, the fracture toughness goes up. But also I have to mention that the crack propagation can be delayed with optimum smaller size of the nanoparticle. Yeah, so this is our facility. It's called the Keck Center for Advanced Microscopy and Microanalysis. So we do a lot of microscopies. We have CM, TM, different AFMs. And we have the supercomputing facilities that we use for atomistic simulations. And also we do kinetic Monte Carlo and FEM. And yeah, so this is our team. The entire team, we are having some bad time because of the COVID. I'm in my office right now, but if I go out, I have to wear masks and PPEs and everything. We're still maintaining that, though almost the entire US is kind of going out without masks. Yeah, thank you, and I'll take any question if you have. Thank you.
Ready to do this presentation. So the focus of the presentation is on our sub-zero pressure machine. It's a new advanced manufacturing technology that we will utilize to mass produce flat and 3D solar cells. And the technology for the solar cells is essentially based off, it's bio-inspired, it's based off the macro of a fly's eye and the nanostructures of a butterfly's wings. So there's actually a Southeast Asian butterfly that has evolved over the millions of years to create these little nanostructures on its wings so it can effectively absorb sunlight at high angles of light incidence. So in something in physics with silicon, if you put some silicon solar cells in a 3D format, there is something called the angle of light incidence that affects how much power it can generate. But essentially, this new butterfly, or actually old butterfly, our technology can imprint these structures in glass and other advanced materials to make different various 3D solar designs. Now, this is also another advanced manufacturing technology that we have in our portfolio. Xolo Germany, our sister company over in Germany, volumetric 3D printed this substrate in 60 seconds or less. If we can't print it in 60 seconds or less, then I don't know who can. So essentially, this new volumetric 3D printing technology overcomes the layer by layer 3D printing process. Now, this is the machine that we're here today to really discuss. And so it's a sub-zero pressure machine. What we essentially do is we take the resin that's typically used in 3D printing. It's a UV curable or photo curable resin that uses a photo initiator to initiate the polymerization of the product. And so what we do is we have a company like Glassamer, for instance. They take fused silica nanoparticles and they actually make it into a UV curable resin that you can 3D print with. And what we did is we saw how difficult these advanced materials were to 3D print. They're pretty much impossible. Like when Glassamer, for instance, sent me the resin, they weren't sending me their best. They sent me their worst. And the reality was we were still able to make 3D shapes with this machine right here. So essentially what we do is we 3D printed a very advanced bit like this, and you can't make it with CNC injection mold and other processes. And then what we do is we attach that bit to a vertical punching pressure machine. And once the resin that we use in 3D printing is frozen below sub-zero temperatures, we're actually able to just punch the 3D shape right in the UV curable resin, creating very, very, very uniform 3D structures that you see in here. This is fused silica nanoparticles that we actually sub-zero imprinted. So not only can we imprint objects, we can also mold them at sub-zero temperatures. So this fused silica glass 3D solar cell substrate actually has a cell wall thickness of 50 microns. And in between each little hexagonal area, the cell wall is only 10 microns. So I don't know if you're too familiar with how breakthrough that is, but it's pretty astonishing. So it actually makes each little subcell flexible. So you can actually flex it left to right. Now, what's interesting about this technology is it's material agnostic. So in about 10 days, we're going to do something similar with silicon particles, silicon carbide particles, and stabilized cubic or stabilized 3 mole zirconia particles. Now, here is some examples of my work. We'll start on the bottom left. So this is actually flexible glass. You can see it's a very small piece. It's only 20 microns. So if you're looking to reduce the balance of system costs in solar, this is the way to do it. This is an example of the particles being frozen in a very, very typical candy mold. And once the particles come out and you get it to a certain temperature, you're able to imprint those 3D shapes in the design. And what is so awesome about this, there's something called the depth of light curing. So we use light, after it's frozen, we use light to set the shape. Now, as you can see in this design, it's not optically transparent. So it ends up that the 3D structure only comes out about 50 microns cell wall thickness. So if you look at the leaders in the industry, Dow Corning, they have 100 micron flat sheet of glass, and Schott in Germany, they typically have 25 to 50 microns. And from my knowledge, none of them can form these advanced 3D shapes because they do it at hot temperatures, and glass has a very honey consistency at hot temperatures. Now at cold temperatures, things change, and we're able to do this. As you can see on the bottom left here, that is a piece that's actually optically transparent. So instead of it polymerizing as a very thin sheet, it's actually a very thick part. And we actually were able to polymerize that in 7 seconds, which is quite fast. Now, on the top right here, this is silicon carbide. It's a cubic silicon carbide that, according to my knowledge, can only be made in Sweden. It's the purest of the silicon carbide. It's a very wide band gap semiconductor. And so our next goal is to form this material into very thin and 3D sheets to potentially see if it has some potential in solar. Silicon carbide battery anodes. There's quite a few industries that have some really advanced technology, but the limitations of their technology lies in the fact that they apply thin film chemical vapor deposition layers of the material. So those in thin film tend to not hold up as long as something where you can actually press, say, a 50 micron sheet or 3D sheet for an anode in a silicon or a silicon carbide like this. Now that will actually increase your yield with our technology. And then on the top right, you can see an example that I did where essentially, if we had the capital, we could take an ASML lithography equipment and we could make a metal or a quartz mold with these butterfly nanostructures. And then we could essentially just punch press those butterfly nanostructures in any material. This machine is material agnostic. So this really opens up the research field where people say, hey, well, I'd be interested in testing this material out. And normally there's nobody that can form it or print it. And we just so happen to do that. Now, this part on the bottom right is something that was volumetric 3D printed. So like I said, I have some IP for some volumetric 3D printing, and Xolo over in Germany has been utilizing that IP and been quite successful. Here's a couple more pictures. Now, this you might find quite interesting. So this is a razor blade. These are micro 3D solar cell substrate, one solar cell substrate. We had multiple of these designs turned into 3D solar cells with the world leader in nanoelectronics and solar thin film, IMEC Belgium. As you can see here, the perovskite was annealed on the 3D substrate. Now, Boston Microfabrication, they're a very advanced micro 3D printing company who is making some serious strides doing things that people say are not possible. This initial evaluation failed. And the reason it failed is because the typical 3D printing process has like a layer by layer process of building a design. Well, that layer by layer process puts constraints on the material we printed. A long story short, the 3D surfaces on this design were not optically smooth enough to make working solar devices. So this initial evaluation failed, but fail for me is just first attempt in learning. So backtracking, essentially the failure that we had with Boston microfabrication with the 3D substrates not having smooth surfaces, we were able to overcome that with this sub-zero pressure machine. Now, this is the machine that's the main focus on my proposal here. So this actually has the way to be thousands of times faster than a conventional 3D printer. So this essentially means we now have the technology to scale the mass manufacturing of these 3D solar cells. And it just so happens that even though we failed with IMEC, because the Boston microfabrication piece did not have smooth surfaces, what we did find out is, wow, this is one of our micro 3D, it's a bifacial micro 3D solar designs. And believe me, I was excited about this because I was told we couldn't do this, and then they did it. So this is, these are only 200 by 200 microns and 600 microns tall. So the XYZ is 200 micron times 200 microns times 600. So you could see the capabilities that IMEC has, and you can also see this advanced technology, the way I shape these structures, how the light concentrates on the image here. So this essentially gives the design to have the ability to absorb sunlight on the exterior volume of the design and the interior volume, which is essentially an industry first for a micro 3D bifacial solar design. And we did have very conformal layers. So this gets me excited because we did fail, but for me, a failure is actually just the next step to getting a win. And so essentially, I'm excited about that. This is the layer stack process that we used for the fabrication of the perovskite micro 3D solar cells. And that's essentially all I have. If anybody has any questions on our technology, I'd be more than happy to answer.
It's very interesting to know that in the first starting of your talk, you were explaining about the shape, butterfly shape.
Those kind words, and yeah, I'm actually quite impressed by all the talks that we heard today. We have a pretty broad variety of topics that we could look into, everything from cement to alloys and viruses and bio compatible materials as well as biosurfaces. So I'm looking forward to the continuation of the conference tomorrow, and I suspect actually that we all do, and that is a very good sign. So let's talk again tomorrow, and yeah, have a good night, and yeah, see you tomorrow.